Thanks so much for joining us today for our first event in our Microphysiological Systems World Summit series. This event today is virtual only, lasting only 4 hours, and our theme is Towards Regulatory Acceptance. Our next event in this series will take place on December 9th, and the theme is Systems Engineering of MPS. And finally, we're looking forward to the big event, and we hope that you'll join us. It's hybrid, virtual, and in person in New Orleans from May 30th to June 3rd of 2022. I'm Elizabeth Baker with the Physicians Committee for Responsible Medicine, representing the American Society for Cellular and Computational Toxicology, and I will be moving us through the event today. For just a quick overview, we're going to start with a keynote address that will be followed by a Q&A. We'll then move into 2 rounds of short presentations that will be 15 minutes each with 5 minutes of Q&A. We'll have a quick bio break in between those, and then we'll wrap up the day with a panel discussion with international regulators, which will then also open up for Q&A. In addition to the presentations today, you have received in your email a booklet of posters for the event. Please do take a look at those. Additionally, we're asking for your help in determining which young investigator will receive an award for their poster that will cover their registration at the 2022 event, as well as some travel funds. Camilla will provide the link in the chat box below, and she'll also be our magician behind the scenes, making sure things run smoothly today and helping with any technical issues. Please do use the Q&A feature to submit your questions as they come to mind. We'll use these for a live Q&A that will be moderated by Lena Smirnova and Ricardo Barrie later in the day. If your question is not answered in the Q&A, we've asked the presenters to answer directly in the Q&A box after their presentations. A big thank you to all the speakers and panelists, as well as the nearly 70 people that contributed to this event for today. From our robust steering committee to an active scientific advisory committee to our virtual events committee that you see listed here, who volunteered a lot of their time to make this event happen. One quick note that at the bottom of your screen, you'll see an option for subtitles. It says CC Live Transcript. Go ahead and click on that and turn on your subtitles if you are interested. Okay, so now I will pass the mic to Thomas Hartung of Center for Alternatives to Animal Testing. Thomas, you have the floor.
Okay, hello. It's exciting times. The disruptive technologies which have shaped the way we're doing cell culture really make technological developments possible we could not dream of some 10 years ago. We are seeing that stem cell technologies, that bioengineering has made it possible to reconstruct organ functionalities and organ architecture in cell culture as we could not imagine. The FDA has coined these microphysiological systems, and these systems, spearheaded by the pharmaceutical industry and the technology providers, are taking over, not only in drug development, but also in safety testing and in basic sciences in general. We see mushrooming of protocols. Essentially every organ can nowadays somehow be bioengineered. We feel that this is time for coming together in an international way, forming a society internationally and nationally in order to allow the synergy of all of the groups involved, help each other to standardize, share experiences, and make this revolution of cell culture actually take place. It is time to get rid of the pan-fried eggs in a petri dish, which look like sunny side up cooked eggs. We really can do better. We can add relevance to what we are doing by allowing physiology to take happen. And then the next step, and this is what the exciting developments in recent years are, to not only do microphysiology, but also micropathophysiology. The disease models, all the things the life scientists, the biomedicine researchers want to know, this is going to happen. I'm excited that we were able to get almost 40 organizations behind this project of a World Summit. We are planning to make this an annual event, and after the one in 2022 in New Orleans, we are planning to move to Europe 1 year later, and this is already in its making. The first World Summit will be co-hosted by Susie Fitzpatrick, our keynote speaker today, by Don Ingber from the Harvard Witness Institute and myself. And we welcome you to this official kickoff for the series of events. And we hope you stay tuned, and we will see you in New Orleans, either in person or virtually. I wish this event today a great start, and thank all of the people involved who made this possible. Thanks a lot.
Okay, thank you very much, Thomas. So for our keynote address, we have Susie Fitzpatrick to tell us about all the exciting things happening at FDA. Susie, you have the floor.
Thank you very much for inviting me here today to talk and to talk about advancing alternatives at FDA. Next slide. First, I want to assure everyone that FDA does encourage the use of new testing methods. We encourage you to come in and talk to us. We are also making sure that our regulators are familiar with these techniques, and that any technology considered for regulatory use has to be proven to be robust, reliable, fit for the context of use, all of which I'll talk about in my next slide. So next slide. This FDA brought some of these concepts to everyone in our FDA roadmap, which was a framework for incorporating new predictive toxicology methods, including MPS. Two things I want to really mention here is that we wanted to have constant communication with our stakeholders, all of you here today. This is not something FDA is doing alone, it's doing in partnership with our stakeholders, and that we believe that research should start with the context of use. In other words, for research that could help advance public health, we suggest starting with the context of use and then moving forward. Therefore that any research in this regulator should be first and should be a partner in all research to help it advance. So next slide. Just quickly, these same themes were put in the ICFAM roadmap, which came out shortly after our predictive roadmap. In the next slide. These themes were also in the TOX 21 strategic and operational plan, communication with our stakeholders, get regulators up front and constantly communicate. So next slide. So we in the government have not only 3 R's that we work with, but 3 C's that run through all these roadmaps and everything that we do in the federal government. Communication with all of you, collaboration, collaborating with all of you to advance these really exciting methodologies, and commitment. We are committed as federal agencies to move forward with incorporating new alternatives into the regulatory schemes. So next slide, please. Where is FDA now? We have an advancing alternative methods page. This is a public page, and if you just type in FDA alternatives into Google, you'll arrive at it. That's Donna's little sketch up front. And then on this, we're putting all papers, presentations, any information on what we're doing on alternatives. And you can also talk to us at alternatives@fda.hhs.gov or any one of our emails. So we're open and excited to talk to all of our stakeholders about advancing all alternatives, especially MPS. So next slide. We also put out an FDA alternatives report. This is on our website. It gives you sort of a highlight of some of the things that we're doing in alternatives. It's not everything, but it's a highlight. And then from now on, we're going to commit to putting it on our website. Recently, ICFAM put out a metrics report where it talked about how each federal agency is going to communicate its advances in alternatives. And one of the things that FDA committed is to put it on our website. And so we're trying very hard to keep this current so that everyone knows what we're doing, and then to allow people to communicate with us through our email address. Next slide, please. What else have we done? We have put together an alternative methods working group under the Office of the Chief Scientist. Donna Mendrick and I co-chair this. This is a cross-agency working group. There are 6 product centers at FDA: foods, vet drugs, pharmaceuticals, human pharmaceuticals, biologics, devices, and tobacco. So we all work together as one agency to discuss alternatives for use in toxicity and efficacy, to interact with our federal and global partners, all of you, as we discuss regulatory performance criteria, and as I said before, to keep updates going on our alternative website. Next slide, please. So what's our first case study? We want to see if this works, that we can all talk together in it, and it's in vitro microphysiological systems. And what have we been asked to do by the Office of the Commissioner? To come up with some terminology for MTS. So, you know, when you see FDA use the terms MPS and organs on a chip, this is what we mean. To identify partners to advance MPS, partners like we hear today with CAAT and the MPS consortium that they're putting together, and other partners, and to draft performance criteria, to discuss these with our stakeholders, and to develop other mechanisms to talk as a community on MPS, to work together to advance this very exciting technology into regulatory use. Next slide, please. So what's our involvement in MPS? Well, FDA in 2010, actually, Dan Tagley and I, through the FDA and NIH Common Fund, awarded grant money to the VIS to develop a heart-lung magnet machine. And this is the first time I remember seeing it at FDA. And Dan too, it was an exciting technology. So in 2011, DARPA came into the FDA and asked us if we would develop a human body on a chip for medical countermeasures. So, and they came in with, Barry Pilato came in with a picture of a box and a funnel going into the box and an arrow coming out. And skeptical though we were in the Office of the Commissioner, we decided to move forward with this exciting project with DARPA, and then NCATS joined that project, and we funded the Tissue Development Program Center together, and that's, the rest is history. It's been a really exciting program and a really exciting journey, and it's amazing that from just 10 years ago, it was just an idea, and now look where we are. But I think one of the lessons, at least FDA learned, it was critical to have regulators at the table from the very beginning if the aim is to be used in a regulatory use. And this is an important lesson that we communicated to ICFAM and our Tox 21 partners, and we hope that we are being successful in communicating that to you. Next slide, please. So we were told to develop definitions, and these are working definitions for microphysiological systems. I won't read them to you, but I will, they're on our website, and we welcome your comments on it and any other definitions that you think that FDA should be developing, so that it's very clear what we're talking about when we talk as an agency. You can communicate with us as alternatives at fda.hhs.gov, or you can communicate with me or Donna Mendrick or any member of our alternatives committee to talk about what you think about these definitions. We're looking at a definition for NAMs at FDA, an FDA definition for that, and we're looking at other definitions too. So next slide. Now, one of the things that we've done is, I've got to move my little people communicate, is one of the things that we wanted to do was to bring in organs on a chip in our laboratories, not only for our different laboratories at FDA to work on organs on a chip, but also to help us as we look at performance criteria. And one of our exciting labs is the CEDAR lab, that's the drug lab, and Alex Ribeiro. And he loaned me some slides here today to show you some of the exciting work that he's doing. If you have any questions on it, I can probably try to answer, but I'm not the expert that he is. He's done a really exciting job of moving this technology forward for CEDAR. So next slide, please. So the CEDAR DARS Labs is looking at evaluating the potential of these systems for use in drug development, obviously. They're looking at toxicity, transport, metabolism, communication, and they have criteria for the initial looking evaluations to look at robustness, reproducibility, and improve the gold standard. As you know, reproducibility was also one of the themes in the NCATS testing centers too, and we look forward to talking to them about some of their criteria too. And they're also looking at, and I can't even see the end of the slide, looking at their preferred published systems and preferentially used in different laboratories and also with cells. So next slide, please. And what are they looking at? They're comparing the liver lobule with the liver chips and they have the CN Bio liver chip. They're looking at long-lasting hepatic properties in both, heterogeneity of the cell types, universality, reliable reproduced methods, and they're looking at to predict chronic toxicity, long-term effects, and model multiple and long-term dosing. So hard to read this because the subtitles are in the way, but next slide, please. This is their general criteria for evaluating the cellular microphysiological systems. They're looking at site-to-site variability, chip-to-chip variability, and they're focusing on PTMS protocols, preparation, treatment, and measurement schedules. And last slide. And this is a roadmap of how they're doing the liver chip characterization. Milestone 1 is assembling, operating an assay of the liver chip, looking at variability endpoints and setting methodological standards. Then they're evaluating different applications of the chip, phase 1 and phase 2 metabolism, accumulation, toxicity, and low absorption. And I think there's a milestone 3, but I can't really see this on the slide set I'm seeing. But I'm sure these slides will be available. Oh, go back, go on. Another really good, really exciting researcher is Dr. Sung in our CBER lab, and she's looking at organs on a chip for regenerative medicine cellular therapy. So she's looking at the process of replacing or regenerating human cells, tissues, or organs to restore or establish normal function. And then you can see some of the things that they're doing in their lab too. And anytime, any papers that either one of these researchers or some of our other exciting research that have this will be published on our website. So next slide. We have entered in a new CRADA with Emulate to bring chips into several of our labs. We brought them into the CVM lab to look at antimicrobial resistance in the intestine on a chip. We have 2 chips at NCTR, and Donna can correct me, one the liver and one the brain on a chip to look at those. And we're bringing the lung chip into the CEDAR lab. And in the CBER lab, we've given money to Dr. Wang to look at COVID with the lung chip and understanding the protective immunity against COVID-19. And so that all of these researchers are just gearing up, and I anticipate we're going to have some exciting results from that. So next slide. Through our medical countermeasures program, we've also awarded a contract to the University of Liverpool and global partners to understand the coronavirus disease severity through the analysis of non-clinical or clinical samples. And this project includes development of in vitro coronavirus models to inform medical countermeasure development and evaluation. So that's another exciting project that we're doing in collaboration with our global partners. So next slide, please. Remember, FDA does a context of use qualification. Context of use is a clearly articulated description delineating the manner, approach, or use of a method. So we're no longer doing the one-size-fits-all type validation that was so long being done with ICVAM and other agencies, where you'd come in with a context of use, how it might be used in a regulatory project, how it might answer a regulatory gap, and then we qualify it. And qualify it is a conclusion that the results of the assessment using this model or assay can be relied upon to have a specific interpretation application in product development and regulatory decision-making. That doesn't mean your tool can't be used for other things, but if it's used for something else, you would have to bring in the underlying data to show that it meets the qualification criteria, performance criteria. So next slide. So what does this really mean in practical terms? Context of use is sort of the key to deciding how much qualification or validation is needed for a particular assay. So if it's a screen in my mind, it's not going to need a lot of qualification. You're going to accept uncertainty, a lot of uncertainty if it's a screen versus whether it's replacing a pivotal study where the consequences of a wrong answer are much greater. So knowing what the context of use is, deciding it will help us define the applicability domain, the limitations. And as I said before, the context can be expanded over time, but coming in with one and starting with that is really what we're suggesting. And then the choice of the reference compounds really depends on what the context of use is. So just coming from a library is one thing, but actually gearing your positive and negative controls to what the context of use is helps to really understand whether this can be qualified. So next step. So these are both of these slides I took from Paul Brown. So these are some of the questions that we would ask, and we're looking at as an agency and what kind of data we would need to answer them. And I think you can see that they're no surprise. Does the assay provide the following questions? Does it mature? What about the cells? Has scientific validity been shown? Reproducible? The applicability being sensitivity, specificity. But I think the most important and the one that we really need to concentrate on is what endpoints are being measured and can you translate them to human use? What is the gold standard that we should be using now? Do we go back to animals for organs on a chip? Do we need organ on chip animals, or do we go forward to human in vivo data? I think this is really, in my opinion, as an alternative person, this is the question that I think is one of the stumbling blocks of moving any alternative forward, is trying to decide what the gold standard should be. And this is something that FDA will be looking at in collaboration with its partners. Next slide. We encourage dialogue with FDA on potential use of MPS and other new predictive text methodologies. Dialogue's important. Come in and talk to us. It's not the same as it was years ago where we were on one side of the table and you guys were on another side of the table. We're all working together, and we have many venues. We have the Alternative Methods Workgroup webinars. So if you go to the FDA Alternatives page, you see that we offer webinars to potential chip developers that have developed a chip and want to present it to the FDA. You can come present it to the FDA, tell us what you think the context of use might be, and we'll have a seminar to all of FDA. We can't guarantee that we're going to qualify it, but for some of these, we've had then a lot of our product centers. You can see we have chips in a lot of our product centers in our labs, and they might start working with you on it. This is how we really started working with Emulate. We went to the really great yearly, every other year, whatever the NCATS meetings that they had, and Emulate suggested that they would like a CRADA with one of the federal agencies, and we took them up on that offer. So getting out there, talking about what you want and helping us. We're also talking to some of the Europeans on the new EU, the follow-up to the EU tox risk and what we can work with them. We have meetings like the National Academy of Science and Energy engineering meeting that we did. Dan Tagley was one of the co-chairs on that. I was on that, and that was a great meeting. All of the slide, it was on microphysiological systems, going from humans to animal research, and that is all available on YouTube. We have other meetings on MPS. As I said, you could talk to us with alternatives. There's pre-IND or IDE meetings. There's other meetings with other parts of the regulatory agencies. There's critical path innovation meetings, which are outside of the application. And there's a lot of other hosts of ways you can discuss with FDA what you're doing with your work and what you think we should be doing. We welcome your input. And last slide. So remember that change takes time, but if we all work together, it will happen. It's amazing that the criteria, and I guess somehow that slide didn't get in here. The criteria, if you looked at the OECD 34 or the ICFAM validation, the criteria hasn't really changed much. We know what it is, but we need to define it and apply it for MPS. So thank you very much for listening to a brief overview of what's happening at FDA. And I encourage you to follow up on our website, on our email, and to all of us if you want to continue this dialogue. Thank you.
Thank you, Susie, very much. I have a couple of questions to you already popping up on the Q&A session. Great presentation and overview. The first question is from anonymous attendees. Thank you for the talk, very clear and comprehensive. It gives the right direction. Great work at FDA. Kudos. Is there a formal validation process? And if so, is it a collaborative approach?
Well, there is a validation process in both. CEDAR, if you look on it, they have a new program called iSTAND too, that's on there. And then CDRH has a qualification program too. Those are two centers that have formal qualification programs. Others of us are talking about what we should be doing to qualify, but CEDAR definitely has one for years. It was started out as a biomarker qualification. They qualified several renal biomarkers for non-clinical testing, and now we're looking at clinical testing and advancing to, like I said, iSTAND and the other partnership. And then CDRH has several qualified tools that they've done.
Okay, thank you. So the next question is coming from Robert Gum. Would you say that MPS is at the screening stage now, or are they replacement users now?
Well, CIPS, FDA and the Alternatives Methods Working Group is working very hard with the IQ Consortium MPS affiliate to have joint seminars between the two of us on where we are with MPS. And I know Paul has looked at what has been submitted so far and hasn't seen any MPS in any of the applications that they've had. We haven't seen it in my center, but I think it's probably, we estimate it's probably being used in the drug development, trying to screen out applicants to go forward, which, so in the early stages, which we wouldn't get that data is not submitted to FDA. So we're trying to see where each of them is at. And of course, if anyone has submitted something, we would like to know about that too. But I think right now it's my guess it would still be in screening, but as anything else, it moves forward. As you get more comfortable with any technology, I mean, remember when we first got computers and we didn't want to use them, and now we can't live without them, or phones, it's the same thing. As we get more comfortable, and that's why we want to make sure our regulators are more comfortable with new technologies before they see them, then it's easier to get them applied.
Okay.
And seminars like this will help. I mean, programs like this that CAAT has put together will really help with the federal agency doing this.
Okay, so we have more time for, and we have a lot of questions. The next one is from Tanvi Shroff. He would like to know, or she, whether it's a list of criteria already accessible to MPS developers from FDA that they can keep in mind when designing the new MPS model.
So I listed some questions, and those I got from Paul Brown and CEDAR about, but they're questions that anyone would ask about feeling comfortable with the data that's coming from MPS, okay, or any alternative. There is, they looked at, the ICVAM had a validation strategy that they put out in, I think, 1997. So we're working through the federal agencies to update that validation strategy and put out something that can be a little bit more informative. So I don't really think that, like I said, I think the relevance question is the one that's the biggest that maybe we need to talk about, and maybe we can explore that in further MPS things about what you need to show validity to human in vivo data, if that's what you're using it for. Other than that, I think the criteria have been the same, and how we interpret them is what we all need to discuss.
Yeah. So then it's a quite tricky question from Cliff Roper. So you might also, I maybe leave it for you to open in writing, but if you want to give a more extended answer, because we have quite some more questions. But this is about the standards. The concern is that we don't have to reach the quite good standards with in vivo experiments. And is that of clinical relevance? It is the indicate that the expectation on MPS in vitro higher than on for the animal models. And does the FDA have a different standards for MPS and in vivo?
Well, I don't think FDA has a standard for them. I think with animal studies, we sort of, at least in the past, took for granted that the data from animal studies were in general relevant to humans. And that was something that started a long time ago, actually, in FDA, because we did some of the early work in toxicology. But I think that with any new alternatives, it is a question because we just don't have the experience with them. And that's why I think one of the things we're looking at, and we haven't, made any decisions on this, what should and would be the gold standard. I think that's something that, whereas the regulatory agencies might talk about, it's something that I think we all need to agree on or discuss.
Yeah, wouldn't be that a bottleneck for the in vitro models on MPS if those questions were not answered for the in vivo models?
Well,
do you think
is it a bottleneck? I mean, I'm sure it's
or obstacle.
It's not an obstacle. It's something we need to, any obstacle can be overcome if we all start discussing on it. And I'm sure that Dan can say more about this since he's doing a lot of excellent work at NCATs to move these things forward. And but it's anything new has to be accepted. It's
Yeah, I think Cliff posted a great question for our panel discussion as well.
Right,
I think,
right.
And
I would be interested in what the panel says on it too.
Yeah. So the next question, we have a couple of minutes. Should we develop organ-on-chip models from animals? This question we discussed many times already, I think, in many different groups.
Should we develop chip models from animals?
Animals, to compare them with animal data.
That is something that could be discussed if that's what they think we need to have certainty about the human data. Of course, we also at our vet drug people are developing animal models for animal veterinary pharmaceuticals, and hopefully maybe a dog on a chip to replace some of the dog studies for target animal safety that we do. And I think having animal models is also important to our medical countermeasure program, and we saw that with COVID, and can we get that was the original reason that we entered into the DARPA agreement for organs on a body on a chip, was to have alternatives under the animal rule for medical countermeasures. So that's also something that's being discussed and is moving forward.
Okay, so I probably have time to two more questions. From Laurie Curley, can the FDA help provide feedback on reference compounds in a given context of use program?
I think if you come in, I'm not into CEDAR I would suggest any new alternative you come into the program or the center that you want to submit that to, and that's early before you start your testing, and that's one of the questions you ask them is what kind of reference comments could you use. I know when we had the first BAA conference on the DARPA Human on a Chip program, we brought in our mostly CEDAR farm tox regulators, and if a person was talking about liver on a chip, they said these are our regulatory questions, these are our regulatory reference compounds we think you might want to use. So these are the things that you should discuss with whatever center you in. Probably you're thinking about CEDAR when you come in to talk to them at the pre-IND meeting or at any other meeting, to talk about what they would want to see, how many they would want to see for a particular context of use.
Okay, so the last question, please, one sentence answer, because this will be time.
I'll try to be brief.
But this is a tricky one. Thank you for the talk. Do you think regulatory approval is realistic? How long it's expected to take?
I think eventually we'll be using these. How long? I don't know. You know, I wish I did, but if I say, oh, it'll be 5 years, so you'll be holding me to it. So I don't think so. I think I'm hoping that we'll do it, and I know that we're all working towards it, and with partners like CAAT and NCATs, and we will get there eventually.
Great, thank you. Okay, I will pass to Elizabeth to announce next speaker. Susie, please, there is a lot of questions in a Q&A box. Please go ahead and type the answers if you can.
Okay.
Over the next presentations.
Thank you for
very
inviting
much
me here today.
Thank you.
Okay, thank you so much, Susie and Lena. We're now moving on to our short talks. So the first theme is standardization, scaling up, and quality control of platforms and cell sources. And we will start with Peter Yanamark of AstraZeneca.
Yeah, so hello.
Okay, your slides are looking good. Thank you.
Thanks. And thanks for inviting me to speak at this summit. It's a great pleasure. I'm a principal scientist in mathematical modeling at AstraZeneca in Sweden. And I will present some work that we do together with the company Tissues in Germany and with the Linköping University in Sweden on disease models in the MPS system. So I work in the disease area, cardiovascular, metabolic, and renal diseases within AstraZeneca. So we are interested in those diseases. And as you may know, cardiovascular diseases are the #1 cause of death globally. And one part of this is insulin resistance that deals with organs like liver and the pancreas. So when initiating this MPS activity, we decided to look at those two organs in the beginning and to look at the interplay between two organs, so try to fit two organs in the same chip. And the basic fundamental question was if there would be a benefit of having the two organs in the very same chip with potential interplay. And we collaborate with Tissues that have their own platform with a chip that has the size approximately of a credit card. And on each chip, there are 2 circuits. And in each circuit, there are 2 wells. And in each well, you put 1 organ. So in this case, we have liver spheroids in 1 well and islets from 1 donor in the other well. And then there's a flow in between, driven by a micropump. So it's a closed system. And this has been published, as you see in the references. So what we found is that there is liver-pancreas crosstalk in this chip. So if we give an insulin bolus dose to this system, so approximately 10 to 11 millimolar at time point 0, you will see a consumption of glucose if you have both organs, both liver and islets, over 48 hours. But if you replace one of the organs with an empty well, you will not have that consumption. And the corresponding insulin curves looks like this. So if you only have the liver, you will get a quite strong insulin peak because there is a lot of glucose. There's a high glucose signal. So insulin goes on a high level. While if you have the 2 organs, the initial insulin that is produced will increase the uptake of glucose mainly by the liver cells. So the glucose concentration goes down, as you see in the left plot, and therefore there's not such a great need for insulin. So this is a quite classic system that has been looked at in the literature for decades. And we collaborate with the Linköping University, with a postdoc called Bellen and Professor Gunnar Södersund. And they are interested in this translation between species in metabolic diseases, and building mechanistic models for that. And the long term, of course, the goal is to replace the animals by microphysiological systems or something similar, to have a direct relationship between the MPS and human. And as Suzanne talked about in the previous talk, this translation is, of course, of great importance. As I said, there's a lot of information about glucose insulin regulation and control in the literature. Our starting point for building a mathematical description of this system is the top model from the literature. It's a dynamic model of glucose insulin and also beta cell mass in man. And this is a long-term model, so it does not only consider one single meal, but it extends over weeks or even months. And as you may know, there is an interplay in such a way that high glucose stimulates insulin production, and insulin in turn stimulates the uptake of glucose by organs. And the beauty of using mathematical modeling is that by a few equations, you can capture several decades of research in this area, and you can use that to interpret your MPS results. So this is the mathematical model of our MPS system. So it consists of two parts, the liver compartment and the islet compartment in blue and red. And in each compartment, we have glucose and we have insulin. And there are also specific descriptions of insulin resistance and the beta cell volume and beta cell secretion capacity. And basically, every arrow here represents some flow of material or some interaction in the system, and they are represented by different terms, mathematical terms in the equations. And for those of you that are a bit interested in mathematics, this is formulated as a set of differential equations. That means that you can simulate what happens over time. So you get a time trajectory in the computer. So what we did, that we constructed an initial model, and this model has both captures the fast response between glucose and insulin, but also slowly occurring processes like insulin resistance and beta cell dynamics. So for instance, that the secretion rate changes over time, and that the beta cell volume changes over time when, for instance, exposed to high glucose. Then we took advantage of data from several experiments conducted both at tissues and at AstraZeneca. And this is an indication of the robustness of the system, because the MPS system could be transferred from tissue to AstraZeneca, and we could basically reproduce similar type of results at both sites. And this is, of course, a very important property of the in vitro system. In the experiments, we considered both co-cultures, where we had both liver cells and pancreas cells, and also single cultures with only liver cells. And we exposed the system to different levels of glucose. Some data were used for calibrate the model, the mathematical model, to find the good parameters for it. And some experiments were left for validation to evaluate the properties of the model. So here you see some typical data from our MPS system. On the x-axis, you have time. You note that it's a 2-week experiment. It's 336 hours. It's a zigzag profile of glucose. And this profile is due to the fact that every 48 hours, the medium is changed. So you get fresh medium, then glucose is consumed during 48 hours, and then you get fresh medium again. During 2 of those 48 hours intervals, we make detailed measures in a so-called glucose tolerance test, a GTT. So in the beginning and in the end of the experiment. The dots in the graph are experimental data, And the line with the cloud around is the model with its uncertainty, the mathematical model. So this is glucose in the co-culture with both organs. And this is glucose concentration in a MPS with only liver. So as you saw before, the glucose consumption is lower in this case because you want to get the insulin response from the islets. And this is the corresponding insulin concentration over time and the corresponding model fit. The main message is that the model, the mathematical model that we have proposed, can be fitted to the data we generate in our MPS system. So now we have both the MPS in vitro system and a corresponding mathematical description. Here are some other model calibration data showing that we can explain different levels of glucose stress to the system. So the blue is basically what you saw before, and the red is more normal glucose levels. So the left plot is glucose and the right plot is insulin. And again, the dots are data and the line is the mathematical model. We also, as I said, we validated the model by testing it on data not used for finding the parameters of the model. And in this case, we used hypoglycemic medium. It's the yellow curve and the yellow dots, and the model could predict those data not used for training purposes. So now the interesting question is, of course, can we use this model to try to make a translation to the human scenario? And if you look at the curves, they don't look really human because you have a slow glucose consumption over 48 hours. If you consider a human being eating a meal, the glucose peaks and goes out over 3 to 4 hours. So the time scales differ a lot between our MPS system and a human being. But since we have the mathematical description, we have a way of reasoning about the translation, because we know that certain parameters in the model are different in the MPS system and in humans. For instance, the volumes differ. So we can start by taking the mathematical model and adapting all the volume parameters to the human scenario. We can also adapt the flow rates, because the flow is different in the MPS and the corresponding blood flow in humans. We can also artificially take into account other organs than the liver that consume glucose. We can artificially add muscles, for instance, and brain that take up a portion of the glucose. So we make those adjustments of the mathematical model. And then we actually end up with curves, simulated curves, the green ones, that look in a relevant order of magnitude compared to data for humans. So the data are from a famous paper by Dalaman from 2007 with data from 200 subjects for a meal response. For glucose, this strategy worked very well. For insulin, we also had to adjust the insulin secretion rate a bit. So apparently, the secretion rate differs between the MPS system and for humans. So this is kind of a proof of concept that in principle, one can have disease models with organ interplay, and one can, by reasoning, scale that to the human scenario. Some aspects of regulatory acceptance of modeling. We know that those data from the Dalaman model, that mathematical model of those data is actually used and approved by FDA as a possible replacement for animal experiments when certifying insulin pumps for type 1 diabetes. And the presenting mathematical model that we use extends this model by taking into account long-term features, and it's based on the top model. As Suzanne said, the regulatory agencies have published modeling guidelines mainly for medical devices like this insulin pump, and for PBPK modeling, so pharmacokinetic modeling. And there's a good overview by Conti from this year. And fundamental requirements for those type of models is that you could validate on independent data not used for model training, and you should predict for similar usage of the model for which it was validated. And that relates to what Suzanne talked about, this context of use. You must apply your model to similar type of questions that was used for the training data.
Peter, you have 1 minute.
Yep. So I would like to acknowledge my collaborators at AstraZeneca, Matrices, and Linköping University. And right now, the team at AstraZeneca, Matrices, and Linköping University are working on a free organ MPS, where we include also the heart in this 2-organ chip. So it's a free organ, and that's the next step. Thanks a lot.
Thank you, Pete, for the exciting talk. I have one question for you in the Q&A session. How to correlate the in vitro MPS data with the in vivo physiological status? I mean that the ratio of liver to islet in the in vitro co-culture system is very different from the in vivo.
Yeah, so that's what I mean. Ideally, when you set up the system, you usually have a factor like 1 to 100,000 in scaling, and you use that factor, of course. But then maybe you have to make other adjustments, like, for instance, as I said, the volume flow, etc. And that can basically only be done in a type of mathematical framework, especially if you also have feedbacks in the system. And that's why we are taking this approach.
Okay, great, thank you. Please watch out the Q&A. I don't see any further questions as for now, but they might come. And if you can type the answers in the Q&A, that would be great during the next presentations. And then we can move to the next speaker, Elizabeth.
Okay, thank you so much to Peter for that presentation, and to Lena. Next up is Camilla Soragni of MIMETAS. Looking good.
You're muted, Camilla.
Okay, sorry, can you hear me?
Yes.
I think so. Okay, so good morning and welcome to this presentation. I'm Camilla Soragni, a PhD student at MIMETAS, and I'm here today to talk about high-throughput phenotypic screening in an organ on a chip, if this is the future or the reality. And to do so, I'm going to present some results, some preliminary results about a robotized compound screen in a perfused 3D microfluidic angiogenesis assay. So before going into these results, I would like to quickly introduce our vision at MIMETAS, and then discuss a little bit about angiogenesis and how is it model, our model that we develop at MIMETAS, and these results. So at MIMETA, we aim to unlock novel biology with better and faster assays in order to have an impact on therapeutic development. And how do we do so? Well, we do thanks to the organoplate, our platform, where we can grow cell culture in a 3D environment under perfusion with direct contact with the ECM. And this is thanks to the organoplate, which has a 384 well format. So it's very compatible with the instruments, the general instruments. And on the back of the plate, we have this glass quality image, glass quality where we have our microfluidic chip. If we look at the structure, this is a sketch, an example, and the design of this microfluidic chip is a 3 channels microfluidic chip, where those channels are separated by this phase guide. So to grow cells, we place the ECM into the middle channel, and then we place the cells on one of the other 2 channels. We let the cells attach to the ECM, because as you can see in this section, there's a direct contact between the ECM and the cells, as the phase guide, which separate the channels, is not along all the height of the channels. Once the cells are attached, thanks to the perfusion realized with organoflow, which is a customized rocker which flip from one angle to the opposite one, and these allow a pump-free perfusion thanks to the gravity. So then this tubule of cells is formed, and thanks to the structure of the plate, the cultures can be easily acquired with a different microscope. And here we have a nice example of some images that shows the different application of the organoplate. Before going into our model of angiogenesis, just recapitulate quickly what is angiogenesis. So angiogenesis is a process where a new vessel is developed from an existing one. Here I place an example with a small tumor, but is also a physiologically situation and healthy situation. So when the small tumor became epoxing and needs new nutrients, start release some angiogenic factors, and these will trigger the sprouts from the original capillary, and then the new vessel is formed. In our body, we have the pro-angiogenic and anti-angiogenic factors, and there must be a balance between them. Otherwise, these can lead to different conditions and different disease. So how do we generally model angiogenesis in vitro? Well, there are different models. There's what is called the scratch model, where you grow cells as a monolayer on generally like a transwell insert, and then you scratch the monolayer and you follow over time the migration of these cells to form again the monolayer. Otherwise, you can seed the ECM and then on top the cells, and those will form this little tubule, and you follow the growth. The third option is placing the cell in droplet of ECM, and those will create this kind of sprouts protrusion-like structure. So those models are quite far away, let's say, for what I just described in the previous slide. There are some limitations. For example, the use of artificial membranes, the lack of perfusion that with the endothelial cells that is a little bit a problem, because they behave differently when there's no perfusion. So generally there's a lack of physiology. They are very far away from the physiologically relevant environment that we can find in vivo. Moreover, they're not easy to image, those kind of models. So if we want to apply, for example, in a screening environment, so the imaging is quite important in order to apply an eye content approach. So what we do at MIMETAS, well, first of all, we start with endothelial cells grown as a tubule, and from there we place the sprouting mix and angiogenic factor on the opposite channel, and these will create a gradient of pro-angiogenic factor, and these will lead to this sprout formation in the ECM channels, as you can see in this nice picture. Here you should see better. There's like the main tubule, and from there all the sprouts that prolong in the ECM channel. From the orthogonal view, we can see that the sprouts are luminized, and from the single plane, from the bottom to the top of the tube channel, we can see that those sprouts are nicely connected and perfusable. So because these models, these angiogenesis models, nicely recapitulate a physiological in vivo situation, and you can see in here where we try different inhibitors, and you can see from the actin images that the assay is capable of capturing different amounts of inhibition. So because of this and the high throughput friendly capability of the platform, we decide to use for the screen. So we decide to screen for protein kinase inhibitors during the sprouts initiation. And to do so, we place together with the sprouting mix the library, the compounds from the library, and the exposure as well as the seeding of ECM and cell has been done in a semi-automated way. Then before starting with exposure, we acquired some phase contrast image of the tubule to use for QC and discharge the tubule that weren't properly formed. After the exposure, we fix and stain the cells with the multi-flow effects, and we stain for the DNA and the actin. And those images were acquired with a confocal microscope and then were used for the analysis. So once the images were acquired, were processed, and the data were organized and filtered for the QC. And then the score starts. So as I already told you, we acquire nuclei, the DNA, and the actin images. So with the nuclei, we evaluate the inhibition, and with the actin image, we evaluate how much this compound has affected the main tubule. So with the nuclei, we focus on the ECM channel, as you can see in here and in this zoom. We localize the cells and we extract the epsilon location. If we place an epsilon axis along here and we place the 0 next to the tubule with cells, on the other side we are at 400 pixels. And then we extract the location of these nuclei, we make the average of the 10 furthest nuclei in this area, we normalize the data, and we quantify the z-score. And this was used to evaluate how much the compound inhibits the sprout formation and growth. With the actin image, we focus our attention on the tubule on this side, and two scientists scored those images from 1 to 4, 1 indicating that the actin stain was completely gone, 4 that the actin was perfectly present. Was scored by 2 scientists, as this data were used later on to train an AI model. Those data were used to see if the compound cause toxicity or disrupt the tubule, and so we call phenotypic score. After scoring those images, we apply those 2 thresholds and we select the hit, and we use those to run the target pathway analysis. So regarding the results, here are a few results where we plot the normalized distance. The distance were normalized against the vehicle control, which is the tubule stimulated with the sprouting mix. Each data point here is a microfluidic chip, and the library was run in duplicate. With no stimulation, we mean the tubule that wasn't exposed and wasn't stimulated with the sprouting mix, and with the positive control, we place the sprouting mix and a compound which inhibits the sprouts. Here we reorganize the data, and we can nicely see that the vehicle control is on the top range, while the no stimulation is on the bottom range of our assay, as it should be, while the positive control is a little bit is closer to the no stimulation, and the sample results nicely fit between the vehicle and positive. in term and the separation between the positive and vehicle control is nicely recapitulated also here with the frequency distribution, where we plot the robust zeta score frequency distribution. So for the hit selection, as I already mentioned, we use two thresholds. We use the robust zeta score below -3 to indicate the inhibition, and here you can see, as well as duplicates, all the results of the compound which pass this threshold. One, we also consider like compound that didn't destroy the main tubule. We discharge quite a lot of them, and we end up with this final hit selection with 54 compounds. Those compounds were used to run some pathway analysis, and in here we have an idea where we plot the robust zeta score and the different target listed in the library. So with this screen, with this proof of concept, we could select multiple potential anti-angiogenesis compounds, and we are verify them with further analysis. So with this study, we demonstrate the feasibility of the sprouting assay for high throughput screening application with a library of 1.5K protein kinase inhibitor compounds, and the image-based phenotypic screening with organoplate offer high resolution information and generates selective hits and novel biological insights. With the data generated with this with this screening, we are still working on some further analysis to see how much data we can extract and what are all the possibility. So to go back to my original question about high throughput phenotypic screening in an organ on a chip, if this is a future or reality, I think that we are in the direction to make it a kind of reality, and I would like to show you this image of a part of the chips acquired during the screen that I think that it gives you an idea that together with the result that I showed you before, is it possible to do it? And with this, I would like to thanks all of our colleagues at MIMETAS, the collaborators, my PhD consortium, iPlacenta, and the Horizon 2020 who found my PhD, and of course the MPC World Summit to invite me to share these interesting results, and thanks also for listening.
Thank you very much, Camilla, for exciting talk. I don't see any questions so far. Maybe some. Now it's coming. First one from Tamara. Thanks for the talk. Is it possible to use an animal-free ECM, no collagen matrix gel?
In general, yes, it's possible. We look a little bit into these alternatives, but yeah, this is something that I think that general the organ on a chip area start looking into that, because of course like ECM and also maybe the serum that is generally used for like the cell culture are still like some kind of controversial situation, because it's not completely animal free. So yeah.
And another follow-up question on ECM again from Tanvi. Have you explored different extracellular matrices of cells from specific donors to potentially create a more donor-specific or disease-specific reaction within your model?
Yes, we tried, but in this situation, for this study, because it was the first application of an organ on a chip technology for screening, we decided to use a very standardized and robust model that we work at MIMETAS from several years. So we decided to use endothelial cells in this setting. But of course, a lot of things are possible, but in order to have a model robust enough in order to apply for this kind of study, we first have to really develop and validate and be sure of the model that we applied. But of course, the next step will be like the use of like disease model or patient relevant model, because that's of course getting closer and closer to what's really happening in vivo.
Thank you. And again, about the cell source, would iPS-derived vascular cells be used? This question from John Paul Sangiovanni.
Yes, this is again is the next step because of course it closed the gap between like the in vitro cell culture and in vivo assays. So it's on the list of things to do for the next steps.
Great. And I have also actually a question. So since your system, what is it was exciting about that you already screened so many compounds, and this of course the next step in the development towards regulatory acceptance. So how do you think what's need to be done from your side to move to the next step and try to get that accepted for the
yeah, I think that this is the first proof of concept. One we are in the way of writing a paper about it with to publish all the data, and of course like following with the secondary screening or confirmation screening, dose response screening, and having so a broader and complete picture of what is to validate also. It's of course the next step, and this will help to confirm that this direction is possible and doable, and the advantage also.
Thank you. I think that's all questions for now.
Okay, thank you very much, Camilla and to Lena. Next up is Monica Piergiovanni of the European Commission Joint Research Center.
Hello, good afternoon, everyone. Let me see. Do you see my screen okay?
Yes, looks good, thank you.
Okay, so thanks for the kind invitation. My name is Monica. I'm working at the Joint Research Center, that is the scientific arm, let's say, of the European Commission. And in particular, I'm working at the ECVAM. So for those of you who don't know us, we are the European Union Reference Laboratory for Alternatives to Animal Testing. And we are mandated by a European directive to work in the validation, research, and dissemination of non-animal methods in science. So briefly, our interests are quite broad. So we, in the sense that we work with many different alternative methods that you see here in the slides. But of course, we are now talking about organ-on-chip, so microphysiological systems. And these technologies entered our radar, I would say 3 to 5 years ago. And as they said, they are getting bigger and bigger every day. So we are, as I was saying, very based in the European framework. And regarding organ-on-chip, we partnered with the European Society for Organ-on-Chip, that's called EUROX. And in the context of regulatory use, our head of unit, that's Maurice Wieland, is heading a regulatory advisory board there. So you may see that we are also from the European side working on on these topics. But enough of that. Let's start our discussion on standards for organ-on-chip. So many international consortia already talked about standards in organ-on-chip and claiming that they will be an enabler to move forwards. But I would briefly like to tell you why. So why are we needing these standards? I also would like to use the 3C, like Suzanne already did from FDA, but we have a different, let's say, acronym for that. So at first, I would say that standards should support characterization of organ-on-chip devices. So there is a common need to have a description of the system, and this will imply a characterization of the components, of course, but also on the recommended operating conditions, the protocols and the SOPs that we need to use the organ-on-chip. Moreover, we would like also to use standards in order to assess the expected performances of the organ-on-chip. So to confirm that the organ-on-chip are, or the devices are following the requirements that they should have. And this will need to follow, of course, both the technical side. So in this sense, I'm talking about fluid flow or pressure, but also the biological parameters. Secondly, I would say that we need standard to compare, and this will need to guarantee that organ-on-chip can be compared with other models, with in vivo, in vitro, or in silico methods. And by using appropriate metrology standard, we will guarantee that we are using the same parameters and the same unit of measurement. That is something that's creating sometimes confusion, at least in some papers that I'm looking to. And the last C, that is the one that's actually the same as the FDA, is the communication. And for the organ-on-chip, I would say that it is important to ensure a proper reporting of the results that are generated, but also of all the aspects of the test methods, because this is something that the regulators will really need to verify and to deeply understand. And this is not only, let's say, going to help towards the regulatory acceptance, but it's going to facilitate understanding among stakeholders in a broader sense. And in this case, the use of a common lexicon and terminology will also increase the confidence in the technology. So I hope that now I convinced you that the word standard for the organ-on-chip can be intended in a very broad sense. So we can include, of course, the formal standards that are those that are mainly developed by international or standardization organizations like, ISO or ASTM. But also we can include other kinds of standardization documents that are best practices, reference materials and guidance documents that are, let's say, usually developed by other kind of agencies or organizations like ICH or OECD. And it's clear now that the use of standards for the organ-on-chip really have multiple roles to advance the field. So at first, standards were traditionally linked to technology, so to the engineering side of the organ-on-chip, let's say. And in this sense, standards can help in bringing innovation to the market, so in helping as SMEs or companies to arrive to the market. And this will help also demonstrating their technological relevance and biological relevance. And finally, I would also say that the use of standards is actually providing basis for regulatory acceptance, because as we saw in our work at ECVAM, but also in other agencies, the use of standardized methods really can help boost the acceptance from the regulators in various fields. That's from the chemical, but also the drug safety. But of course, we are not going to have one standard that's going to make the job for everything. We will need to have multiple standards, each of them that will need to address a specific aspect of the technology that the organ-on-chip is having. And for each aspect, we need to identify a standardization roadmap. So which is the best option that we need to have. And to further advance in this direction, we thought that there is a need to involve really the standardization people, so the standardizers. and to bring them together with the developers and regulators. And for this purpose, we at the Joint Research Center, together with CENELEC, which are the European standardization organizations, we organized this workshop that's part of a series called Putting Science into Standards. And in 2021, it was on organ on chip. And it took place online, unfortunately, at the end of April this year. So the first half day was dedicated to creating a common ground for the participants that, as I was saying, were very multidisciplinary. But the main part of the workshop was actually on the second day, where we had 3 parallel sessions that you see here in the slides. And the participants were able to choose between engineering, life science, regulatory, and data management. And in each track, the discussion was led by a panel of experts, and it was complemented by input from the participants through Slido. And the main point of the track was to identify needs and priorities for each of these areas. So I will try to briefly summarize those, starting from the engineering session that was the most, let's say, related to formal standardization activities. We had these 3 sub-sessions, and one main outcome of all 3 of them was that participants felt that ready-to-use device and components would greatly facilitate the adoption of organ on chip, only if these components are suitably characterized. Moreover, a standard catalog of components and standardized dimension and geometries will help the interoperability of the organ-on-chip devices, but at the same time ensuring enough freedom, let's say, for the designer to design the new organ-on-chip. And finally, a note on the material. So the participants felt that a general standard protocol for material characterization would really help tackle one of the main issues that we have with materials in the organ on chip, which is the uncontrolled absorption of molecules on the PDMS, for instance. Also, the life science track was divided in 3 subsessions that, of course, were very integrated in this sense, in this particular track. And of course, we have to admit that standardizing biology per se is not, of course, possible because of the nature of biology. But all the experts recognize that there is a need to standardize the procedures that we use to model biology. And this in order to ensure relevance, reproducibility, and robustness. So one main recommendation was to make a better use of guidance documents. And in this sense, there was a couple that were overcoming the others, that are the good in vitro method practices, that's called GVIMP, and the good cell culture practices, that they both give indication on how to properly perform the experiments with any device. And finally, for an organ-on-chip model to be fit for its intended purpose, it needs to capture the key aspects of physiology and to reproduce specific organ functions. And of course, this can only be done with a proper cell model that has a functionality that is expected. And here we come in the need to have reliable assays and biomarkers that should be properly qualified, as Suzanne already said. And finally, we had the regulatory and data management track. And here I would say that, of course, all the regulators that were present confirmed that they are looking favorably to the use of organ on chip. And they suggest that the community focuses initially on data-rich context of uses. For instance, that could be liver metabolism and also heart toxicity. This will be, of course, need to be complemented with reference compounds. And the suggestion in the choice of these compounds is that they are commercially available, so that there is a control of the compound that we are using in the experiment, and that they are given in human relevant doses. Another point that is going to, in the opinion of the panelists, promote the regulatory uptake is the adherence to reporting standard that will need to include a description of a minimum set of parameters that can guarantee that the device is working. So for the organ on chip, sometimes there is a feeling that we are not sure if it's working or if it's not working, and if it's working as it should or not. And finally, about data sharing. Of course, this is an important topic also for other methods, other in vitro methods, and in general for a wide range of things in the biological sciences. But some points will be that data should be shared across different communities, and that the panel pointed out that there are a lot of community standards already existing in other fields, like bioinformatics, for instance, that can be also applied in other topics, like for organ on chip, for instance. And this will need to come together with a definition of a common vocabulary, uniform data formats, and a minimum information checklist for metadata, so that when a data is put on a repository, it is clearly labeled and understandable. So to conclude the workshop, we had a plenary panel discussion where we had senior representatives from different fields. You see here that we have standardizers, developers, representatives from pharma, and also policymakers. And they shared their views on how to move forward. So a first outcome was that end users are primarily attracted by off-the-shelf devices that are well characterized. So they will want to have a bottom-up approach that, to summarize it, is give me the plastic and I will put the biology in it. Then there is, on a more practical note, we also have a good proposal from the panel, that is the creation of a Sensenelec focus group, that is a formal experts group inside the standardization agency that will be responsible to define a roadmap and identify priorities in the organ on chip. So this group will take the outcome of the workshop and it will bring it forward into the Sensenelec formal standardization framework. So this focus group should also, of course, interact with the European Society for Organ on Chip, also to provide expertise. And we are now waiting to see how we can actually bring the standardization work forward in this sense. So with this, I thank you. Please reach out to me. You have my contacts here. And if you want to go deeper in the world of standards for organ on chip, please check out this article that is going to be published on Lab on Chip in the next days. Thank you.
Thank you, Monica. It's really great talk, and you pointed out the questions and points exactly need to tackle. There are a couple of questions here for you. The first one is from Itze Pantoja, who is asking, are you considering the 3 Cs relative to other regulatory modalities in other continents?
No, to be honest, that was just a coincidence that we had the 3 Cs. But indeed, when Suzanne was talking about the 3 Cs, then it came up to my mind that we also had the 3 Cs, but for a totally different point. So I think that they are, let's say, general considerations that can be applied for organ on chip, but also for many other methods. So we can call them 3 Cs, or we can call them another way. It's the same. I mean, the basics are more or less shared with all of the international and European agencies.
Okay, thank you. Next question is whether ECVAM works with EMA.
Yes, sure. Of course, we are working with EMA, of course, for the medicines. We are working a lot with ECHA for the chemicals, and with EFSA for the food safety. So we are actually trying to bring our scientific support to all of the agency and directorate generals that contact us. So definitely yes.
So now next is not a question, it's rather a compliment. Thank you, Monica. And I would like to read it loud. I really like that you have highlighted freedom for developers, but also initiate guidance and standards by which they need to achieve. You have also demonstrated the high quality processes and procedures need to be identified and then put into place and identify all stakeholders. I look forward to the paper, and this is from Cliff Roper.
Thanks. Just to say that, of course, standards in the past were perceived as these sort of cages where, okay, we all do screens that have, I don't know, 10 centimeters by 10 centimeters, and that's it. But in the end, that's not the purpose of standard. The purpose of standard, as we say it, is to define the requirements that can be general and characterize those requirements. So freedom of the designers is there for everyone.
Great. So one more question I have for you. Are there any initiatives where a company can join ECVAM in developing guidance and standards?
Of course. We are very open to test method developers, and this is not only for organ on chip, of course, this is for every sort of non-animal methods. So just contact me, you have my address, and if it's an organ on chip related, I will take care of that. Otherwise, I will pass on to other colleagues if it's another argument.
Thank you very much. So I think I covered all the questions which pop up so far. Please watch the Q&A. Maybe some more questions will come to you. And I will pass to our last speaker, Elizabeth, to present our last speaker.
Okay, thank you very much, Monica and Lena. Our next speaker is Anthony Holmes of the NC3Rs.
Hi, thanks very much, Liz. Hopefully you can see my slides if I go to presentation mode.
Yes, looks great. Thank you.
Okay, excellent. Thank you very much. Good morning, good afternoon, everybody. Good evening, wherever you may be joining us from. And thank you to the organisers for this opportunity to speak today. It really is a great delight to be able to tell you about this recently established international working group that we've developed, which is really aimed at supporting the wider adoption of MPS models for COVID research, but which could have impacts on the use of these models for other infectious diseases and potentially for future diseases of pandemic potential as well. And so my intention today is hopefully to engage you in the group and encourage some of you to join us in helping to move these aspirations forward. Because now's the perfect time to get involved, because we're really starting to define what the key priorities are for the group. We'd love to hear your thoughts on this as well. So I don't really need to dwell on this slide or state at any length what the impact of the COVID pandemic has been on the world. You can see from this infographic from the World Health Organization that the scale of the impact on human health and with the number of cases and deaths continuing to rise, and this doesn't take into account the wider health impacts as a result of overcrowded hospitals or reduced access to timely healthcare and emergency treatments. But it's because of this incredible health need that we've seen science move at an unprecedented pace and seen science and scientists thrown under the spotlight like we've never seen before. And it's hard to think about the current pandemic in terms of positives, but if there are positives that come from this, I guess one of them really has to be the way in which scientists have come together at great pace across borders, disciplines and sectors to move discovery forward and unravel the key questions about the virus, understand how it spreads and who's most at risk, and develop tests and drugs and vaccines to tackle it. And knowledge of the characteristics of the virus, including mechanisms of cell entry, replication and transmission, has been essential in developing novel and effective diagnostics, therapeutics and vaccines. And the speed at which scientists have been able to identify the essential features of the virus has really been breathtaking. Animal models have played a significant role in this, and a whole zoo of models have been developed to understand the disease and its transmission in natural and intermediate hosts, but also in several species that have been reported to be susceptible to SARS-CoV-2 either naturally or experimentally. And you can see here in this figure taken from Madi et al. That the variety of species that have been used for these studies experimentally. But we will recognize they have their limitations, and this is something that I'll come back to in my next slide. But what has been really great to see is how collaborative the animal modeling community has been in developing these models. And much of this effort has been coordinated by the World Health Organization, who as part of their R&D blueprint for COVID has established the COVID-19 animal models working group to provide a platform for the sharing of data, reagents, and methods which can help reduce duplication of effort and accelerate the development of new therapies. And this has already resulted in avoiding unnecessary in vivo experimentation and helping to advance the 3Rs. And this group has also opened up their platform for MPS model developers as well to showcase their work. And I believe that there's probably more that can be done here, and this is something that I'll come back to. So despite the advances in knowledge that animal models have provided, they are not without the limitations, and some of which are highlighted here. So the current demand for animal models exceeds capacity for commercial breeders, and this is particularly true of primates and transgenic animals. And this can really have an impact on the speed of progress, but also the verification of results. Limited lethality, and in fact, the majority of models are only capable of recapitulating the mildest forms of the disease, is a major shortcoming. Mice, which are the most commonly used species, are not easily infectable with SARS-CoV-2 due to the differences between human and murine receptors important to the disease. There have been some transgenic mice developed which express human genes and receptors, but again, these tend to show limited infectability for SARS-CoV-2 and develop mild symptoms. But there's also physiological and anatomical differences. So mice have a different airway branching system compared to humans. They have much smaller alveolar unit diameter, and there's also differences in the regenerative pulmonary response. And so to maximize the translational value of these models, it's been suggested that we can increase the use of animals with comorbidities, and used aged animals, use both male and female animals, and also develop more transgenic animals. And all of this result in a significant increase in animal use, but whether this will correlate to advances in knowledge and availability of therapeutics remains to be seen. And here you can see just a few of the references which are available, which really do a great job at looking at the various utility and limitations of the available animal models at the moment. But there is an alternative approach, and that greater use of MPS could be used to address some of these questions and reduce our reliance on animal models, or inform animal model development where it may be necessary. And their potential utility is reflected in the significant number of new models available for assessing the impact of SARS-CoV-2 on the lungs and other organ systems affected by the disease. And now much of this research focuses on the lungs for obvious reasons. And there are lung on a chip and lung organoid models that have been infected with virus and that have been used to reveal important aspects of both infection response and viral replication, including corroborating the dependence for SARS-CoV-2 on ACE2 for cell entry, the role of interferon-associated and pro-inflammatory genes in the innate immune response to virus, and to better understand the kinetics of viral replication. Circulating immune cells have been incorporated in these models to study the interplay between viral infection and immune cell recruitment, and the role that this plays in both barrier injury and exacerbated inflammation. And they've also been used to examine damage to the vasculature within the lungs caused by the virus and the mechanism by which this occurs. We've also seen significant funding in this space, such as this $5 million project supported through the FDA's Medical Countermeasures Initiative, which will link researchers globally to help inform development of SARS-CoV-2 diagnostics and vaccines and therapeutics, and which will compare coronavirus infection in both MPS and animal models and also in humans. And there's also been significant effort to use MPS to understand how the virus affects other organs where ACE2 receptors are prevalent. So for example, there's been the use of brain organoids to examine SARS-CoV-2 neurotrophism, the use of blood vessel and kidney organoids to demonstrate the inhibition of infection by human recombinant soluble ACE2, and gut organoids to demonstrate infection and replication of virus within the small intestine. And all of these studies together really highlight the potential utility of MPS as experimental models. But just as with animal models, such a response to a singular focus on a global scale can risk being fragmented, increasing the chances of duplicating effort and already overstretched resources. And how potential users navigate through the increasing number of models to identify the right ones for their needs is really an important question, given the number of models available and how much is riding on the correct choices being made. So the UK National Centre for the 3Rs and the US validation agency NICETM have together tried to support the research community in its efforts to expedite the adoption of MPS for COVID-19 research through the creation of the MPS for COVID Research Working Group, or the MPS Core. And we're doing this in partnership with a number of individuals that you can see here, who will be well known to many of you. And I'd really like to take this opportunity to thank them for their fantastic support in getting this initiative off the ground and helping to move it forward. And our aim is to try and connect researchers and methods developers, drug and vaccine manufacturers and regulators to collectively maximize the scientific, animal reduction and public health impacts that MPS offer in better understanding and treating COVID-19, and to support their adoption to prepare for potential future diseases with epidemic potential. And we've conceived our working group to deliver 6 key objectives. So firstly, to provide a forum for interaction and engagement between researchers internationally, to raise awareness of available COVID-19 and MPS technologies to support their application in developing novel therapeutics, to understand the regulatory context of use for these models, to provide cross-discipline and sector expertise in characterizing model performance and readiness criteria, to support the assessment of MPS against in vivo and human data, and to ensure the animal reduction opportunities these model platforms offer are also recognized. And so just to give you some details now about the working group. So it's fairly new. We were established in January this year, but we already have more than 100 members from around the world who represent all of the relevant sectors, including academia, industry, both large pharma and smaller biotechs as well, regulators and government agencies that have a direct interest in the development and application of MPS. And that this includes a good mix of both potential MPS end users applying the models for basic biology, understanding disease pathophysiology and in therapeutics development, and also model developers from academia and small companies. And we can see that about 50% of the working group are currently using MPS as part of their SARS-CoV-2 research, with another 20% or so planning to use these devices. So we've got really good experience within the group in terms of how and where these models can be applied for advancing SARS-CoV-2 research. And hopefully this will provide a significant number of case studies that we can use to hopefully build confidence in these approaches and expand their use for COVID, but also for other infectious diseases as well. But data not shown here today suggests that from within the working group suggests that we'll have to wait potentially up to 12 months before this data is available for sharing. But we will do our best to try and expedite this and work with our working group members as much as we can to support that. So one of the things that we wanted to do very early on was to use the working group to try and help define what the priorities should be for this particular activity. So we asked members about both the scientific and practical challenges that they consider to be the most of a barrier to the wider uptake of MPS for COVID research. And so starting with the scientific challenges, you can see that this includes a wide range of challenges, such as the regulatory acceptance of data generated in the models, awareness of models and lack of confidence in the models, but the most significant of these being access to material, including human disease material and well-characterized cell sources, lack of standards or performance criteria to really assess the utility of each model against each other, as highlighted by the previous speaker, and building sufficient complexity into the models with the incorporation of innate and adaptive immune responses. And in terms of the practical challenges, these included the models being difficult to use and requiring specifically trained personnel and the financial outlay required to bring these models in-house. But the major practical challenges were defined as being access to BSL-3 facilities required for the use of virus within these models safely, and perhaps unsurprisingly, with the number of academics that we have on the group, access to appropriate funding. So these responses suggest that multiple factors contribute to the problem. On their own, each of these may be resolvable, but when combined may prove to be quite a challenge. So armed with this knowledge, we're trying to build a program of work that will support the community in overcoming or at least reducing the scale.
Al caffè, buona idea. No.
So I've got some background noise.
Okay.
Could I ask everyone to mute, please?
Okay, thank you. So our initial focus has very much been on sharing models, data and experience amongst the working group members. And central to this has been supporting the expansion of the microphysiology systems database to include a COVID-19 portal. I'm sure many of you are probably aware of the database already, but essentially it's a web-based resource developed by researchers from the University of Pittsburgh and supported by NCATS to accelerate the development and application of MPS for basic research and drug development. And so the database will take MPS data and aggregate this with linked reference data from a number of different sources that you can see here to support the development, application, and interpretation of the models. There's also built-in tools which can help assess the reproducibility and transferability of MPS models, and there are computational models to enhance the value of MPS models for understanding disease mechanisms, compound toxicity, and prediction of drug pharmacokinetics. And so NICETM has partnered with the Microphysiological Systems Database developers to expand the platform to include a COVID-19 disease portal for researchers to share their MPS models used in understanding the disease and developing potential therapeutics. The portal went live in April this year, and it already includes summaries and links to experimental model designs and study components, including reagents, cells, devices, compounds, assays, and assay kits, which can be used to develop COVID-19 in vitro models. It's got links to in vivo and computational model data of COVID-19 and databases of clinical trial data that could inform the design and validation of MPS models. It has a series of user guides to assist with the addition of models, the creation of studies, and uploading data analysis of results. And there's also a permission structure in place that allow users to choose when their data can be viewed by the MPS core group and when it can be released more publicly. And a very important aspect of the portal is that you can find and connect with MPS core members directly as potential collaborators and resources using the private MPS core member profile list. So once you've registered on the platform, MPS core members get the opportunity to create their own profiles, which are visible only to other MPS core members. And this includes information on what platforms you're using or may have an interest in, what species and tissues you're using or may be interested in, various applications of the models, the focus and goals of your SARS-CoV-2 research, and availability of BSL-3 and BSL-4 facilities. So if you haven't already taken a look at the MPSD database, I would strongly encourage you to do so. There's a large number of helpful resources to guide you through its many functions and how you can interact with it to support your MPS research or application. And I'd also just like to highlight that you'll soon be able to access through the MPS core website a recording of a webinar we recently hosted where the database developers described the platform in great detail and where a user described how they have used the database to input their own model data and take advantage of the various modeling tools within it. And to further connect the working group members, we have hosted a number of webinars to raise awareness of available COVID and MPS technologies to support their application in COVID research and drug development. This includes an introductory meeting where we introduce the plans for the working group, showcase some ongoing initiatives in MPS model development and their application to SARS-CoV-2 research, and introduce the MPS database and began discussions with the working group to really understand where we could add value to the community. We followed this up with a workshop in April where and this included an excellent agenda of speakers showcasing opportunities for microphysiological systems applications in therapeutic development and in understanding disease mechanisms, and a really great panel discussion to further explore the challenges and the opportunities that were identified through the survey of the working group members to try and define priorities for the group to focus on. And some of these priorities, including establishing a seminar series to share the latest research findings and model development, which is due to start next week. And you can see here some of the dates for future seminars. And we're still accepting abstracts for these. So, if you have some research finding or some models that could be relevant to COVID and SARS-CoV-2 research, and you'd like to share these, please let us know. But we're also currently collating information on sources of funding, accessible BSL-3 facilities, cell and tissue sources. We're working to try and define performance criteria for these models based on their potential context of use. We're collating information on the 3Rs potential of these models. And we're trying to forge links with the WHO Animal Models Working Group to really maximize the opportunities for collaboration between our various members. And if you want to know more about this work, you can go to the website that you can see here, where you'll also find recordings from the first 2 workshops. So just to quickly summarize, clearly MPS offers significant opportunities to support COVID-19 research and therapeutics development and reduce our reliance on animal models. And the foundations that we put in place for this now could have significant impacts for how these devices are applied in generally, but also in potential future pandemics. The MPS core aims to support these efforts by connecting researchers across sectors and disciplines to maximize the scientific, public health, and 3Rs potential of these models. We are absolutely keen to collaborate. We know that there are others working in this space, and we don't want to duplicate effort, but rather work together to try and add value where we can. But we really do need your expertise and support. So if you're not already part of the MPS core, you'd like to know more about it, you'd like to join us, please do get in touch and let us know. And so I'd just like to thank you for your time, but also just to thank my co-chair of the group, Nicole Kleinstroy, the acting director of NICETM, and the other organising committee members who have been so supportive in helping get this off the ground. Thank you.
Thank you, Anthony, very much. It's so exciting, and there's such a great work you guys doing and putting all those resources together. I would like to ask if you could put in a chat for all attendees some links on, for example, MPS database and the resources that people can click and look. I have a quick question to you. What would be post-pandemic work of this working group? Would you extend that to potential next pandemic or to infectious diseases in general? So what do you have any thoughts on that?
Yeah, I think that's a really good question. And you know, obviously, when post-pandemic is, you know, who knows? And I think even when we are considered to be post-pandemic, there'll still be a lot of work done in this space around COVID and SARS-CoV-2 anyway that we can really try and try and connect and push forward. But I think, you know, one of the aims is that we want to try and support the use of MPS models for future potential epidemics for other infectious diseases. And so I think there'll be great potential to continue this work in some of those other areas that might be up and coming, some of those infectious disease areas that we know exist at the moment where perhaps MPS aren't being applied so fully. So, you know, I think there will be a number of opportunities that we can look to take this work forward. And obviously, as that occurs, we'll look to bring in new expertise and engage with new particular audiences. And a lot of that will be defined and supported by the working group as we go forward.
Okay, thank you. So we have a couple of questions here in the Q&A session. Is NC3Rs accept new concepts MPS to be presented as the part of your innovation platform?
Sorry, can you say that? Repeat that one.
Will the NC3Rs accept new concepts and MPS to be presented as a part of your innovation platform? I think you mentioned something that a little bit in your talk.
So I think this may be directed more about an innovation platform that the NC3Rs has specifically, and forgive me if that's not correct, and hopefully the questioner can ask me directly afterwards. But so NCRs, we have an innovation platform where we are sharing new technologies and approaches relevant to the 3Rs, all of the 3Rs. We would be delighted to hear from researchers that have new models and new concepts that they would like to showcase through that innovation platform. We collaborate with an organization to provide that kind of digital matchmaking service to really showcase some of these new technologies with 3Rs potential. So yes, please do get in touch with us if you have different models and technologies that you think would be fit for that. And we also have an innovation funding program as well, where some of these technologies may be applicable as well.
Thank you. So another question is from Karina Caunalo-Conteras. Excellent talk. Actual manufacturers, plastics, membranes, microfluidics, etc. welcome to join the MPS core to collaborate developing their methods.
Absolutely, absolutely. I mean, we want to be as inclusive as possible. You know, creating these technology platforms in MPS require a lot of expertise and different backgrounds and disciplines and engineering components and everything else. So we would be delighted to hear from researchers, manufacturers, small companies that work in this space that provide, you know, enabling technologies and components, because, you know, there could be different components that current members of the working group aren't aware of that could really try and support the development of something slightly new, modification of a technology which could have an impact. So we do want those to be part of the discussion as well.
Then one question from anonymous attendee. What is your expert opinion on using MPS that rely on whole blood circuit emulating real tissue?
That's a good question, and maybe out with my level of expertise. I wonder if there may actually be researchers or model developers that are within the audience and within the panel, I'm sure, that are perhaps better placed to answer that question directly. And so if it's possible that could be opened up to other panelists, then that would be great.
Okay, so Please, everyone who has the knowledge on that, please post something on Q&A, and then we will make it visible for everyone. Thank you.
Okay, and I could just see as well that Mark Sherdak, who is from the University of Pittsburgh and is behind the development of the microphysiological system database, has just put the website for the database in the chat as well. So go and have a look at that and have a play. It's a fantastic resource.
Yeah, thank you very much. I think that is all questions we have for now.
Thank you.
Any urgent questions from the panelists who can just ask? Okay, we are perfect on time, Elizabeth. Take it from here.
Yes, we're perfect on time. Thank you to all the speakers and presenters, and thank you to Anthony for that talk. We are going to take just a very quick 5-minute break, and we'll be back at 11 a.m. Eastern to get started, and we will start on time. Thank you.
And during the break, we will post the poll to vote for the posters. Please have a look, and if you had a look into the poster book, please vote for the best poster you think was the best for the Young Investigator Award.
Okay, Elizabeth, I'm gonna end the polling. Give it 5 more seconds. If you didn't vote, please vote. And so we can start in time. Okay, I'm closing the polls.
Okay, thank you, Camilla. Welcome back, everyone. We are moving into the second round of the short talks. We'll hear about obstacles to accomplishing regulatory acceptance. And Seiichi Ashida of the National Institute of Health Sciences and Sojo University will start us off.
Okay, so can you see my slides?
Yes, they look good,
Okay.
thank you.
Okay, so it was a great pleasure for me to have this opportunity of presentation. So, title of my talk is MPS Initiatives for Industrial Implementation and Regulatory Acceptance in Japan. My name is Seiichi Ishida from National Institute of Health Sciences and Sojo University. Today, I'm going to report an overview of the AMED MPS project in Japan. So this slide shows the organization chart of the MPS project in Japan conducted by AMED. This project was launched in 2017. This is the 5th year of the project. The annual budget is about $9 million. The project consists of 4 programs. Program 2 is developing and improving MPS devices, and program 4 is establishing the supply of hepatocytes from Japanese population. Program 1 is responsible for the production of MPS devices developed by program 2. Program 3 will standardize the MPS with cells provided by program 1 and 2. Characteristically, 5 pharmaceutical companies and a device supplier are also participating in this project in developing models and conducting standardized tests for MPS. The project is equipped with a central laboratory mainly for the development of medium volume production techniques of MPS and to provide a place for MPS evaluation by pharmaceutical users. Open Laboratory is working on the evaluation of commercially available MPS. Although not shown in the diagram, Pharmaceuticals and Medical Device Agency, PMDA, which undertakes the review of pharmaceuticals in Japan, participate in this project as an observer and exchanges opinions. The MPS project is now in the 5th year, and at this point, 4 types are on the verge of commercialization. Device to evaluate the flow of substances such as the enterohepatic circulation, device to apply shear stress by perfusion of cell culture on the upper and lower side of the membrane, and devices to verify the interaction between 2 organs by connecting them. You can get the information about the detailed future of each device from this URL. The process of regulatory acceptance, which is the topic of this session, requires the participation of 4 stakeholders: academia, supplier companies, and users, mainly pharma and CRO, and regulatory sector. Our project also involves these stakeholders, and since the start of the project, we have had many discussions to deepen our collaboration. It goes without saying that the key is for these stakeholders to work closely with each other. However, discussions have revealed that there are gaps in the understandings and the perception of the development and use of MPS. First of all, there is a big difference in the perception of device usability between academia, device manufacturing suppliers, and users. As you can imagine, original devices developed by academia often require specialized training in device handling before they can be used in actual testing, making it difficult to use them in drug development. On the other hand, from the user's point of view, there are usually limited opportunities to exchange opinions on what kind of device will be developed, and we have experienced that devices that fit the context of use that users want have not been developed, or that applications for devices have not been developed properly. In addition, it is necessary to discuss the extent to which the evaluation of new drug candidates using the MPS device can be applied to new drug review documents between pharmaceutical companies and regulatory authorities. Therefore, in this project, we have taken multiple approaches to fill these gaps. In order to bridge the first gap, we have had a series of close discussion between the labs in academia, the users in the pharmaceutical companies, and the device supply companies. As for the second gap, we are providing the central laboratory for pharmaceutical companies to actively participate in evaluation tests of MPS devices and to discuss their feedback to the supplier companies. Although there were some difficulties due to the COVID-19 pandemic, researchers from university laboratories and pharmaceutical company researchers gather in one laboratory to discuss everything from handling MPS devices to conducting evaluation tests and examine the result face-to-face. As for the third gap, the first thing Program 3 is currently doing is preparing documentation for publication that regulators can refer to based on these experiences accumulated as project progression. Through our approach to building these, bridging these gaps, we believe the following trend can be formed. So support for gap 1 will enable MPS device with improved usability to be offered to users. In addition, by creating a place where user can directly evaluate these devices, we will be able to extend the context of MPS use in pharmaceutical companies. And by documenting them, regulators will have a better understanding of the application of MPS. The following slides show the implementation status of each approach. First, I would like to share a specific example of a cross discussion between an academic laboratory as the developer, a pharmaceutical company as the user, and a supply company. This is a classical Barak type MPS. I dare to show it here because it is easy to understand to think about the composition of MPS. As you all know, MPS is often used to culture cells in a cell culture device. In this case, a glass slide type chip. The most important part of the MPS is the type of cells to be placed here, but it is also necessary to construct a perfusion circuit for supplying medium to culture and maintain cells in these micro spaces. The perfusion circuit is a part that has not been used in conventional static culture, so it is necessary to consider as well. Therefore, we will introduce our effort to improve the usability of cells and the surrounding apparatus. First, the cells. In this presentation, I will introduce an example of hepatocyte for which verification is proceeding ahead of other tissues. The project held a hearing on the performance standards necessary for conducting metabolism and kinetic studies of drugs using hepatocyte. As a result, we have found that users are interested in the cellular functions listed here. The basic functions are the active function expression of phase 1 and phase 2 enzymes and transporters. Therefore, using frozen hepatocyte as a standard, we created a protocol for common culture method and activity evaluation procedures and conducted evaluation in the laboratory of the 6 participating instruction in institutions. This is the result of the evaluation of the activity of 5 major molecule species of drug metabolism enzyme P450. By creating a common protocol, it is possible to evaluate the activity with good relationship, even in different institutes. Next, we will discuss the standard for the element that makes up the perfusion circuit necessary to maintain the cell cultures in the device. Through discussion with users and suppliers, we have developed a lot of issues that arise with materials and equipment when evaluating drugs, especially small molecule drugs. Here we introduce an example of study on adsorption of low molecular weight compounds to device materials. Because of the ease of producing, high observability, and low cost, PDMS has been used extensively in MPS research, but it has also been pointed out that the adsorption is a problem for hydrophobic substances. We are currently evaluating the adsorption of chemicals, especially low molecular weight drugs, not only on PDMS, but also on materials used in MPS devices. As a result, the project has been developing devices with PDMS-free materials. One is the pneumatically driven devices, and the other is small stacker-based perfusion devices. Next, I would like to introduce our effort on how to adapt the devices developed to the user's desired context of use. Unlike conventional culture plates, MPS are complicated to handle. Therefore, we put our effort on how to make it easier for users to use these new culture devices. Program 3 takes the lead in creating a detailed procedure for handling MPS platform. In doing so, we do not just create procedure manuals, but also have them peer-reviewed by experts, aiming to ensure that any users can reproduce the operation. After preparing the cell and devices, as well as their operating procedure, we conducted a device test with user participation. The left side shows each step. The chart on the right shows the duration of each steps in months. After going through the documentation processes described in this previous slide, we conducted a training session with users' participation. We started roughly 6 months on preparation. The training is conducted in a central laboratory. After these preparation periods, we conducted verification tests of the device at each of the users. The verification tests were conducted in accordance with the procedure described earlier. The details of the experiment were recorded and summarized after the completion of the test, along with the result of the experiment. It took about 3 months from the implementation of the verification test to the summarization. The project has been working on the standardization of test method and implementation of users' experience on the use of the device being developed to suit the COU. Based on this experience, we are currently working on documenting the standards and consideration point for the utilization, the new culture system, MPS. The slide shows the chart of the process. The COU of which final regulatory acceptance is required will be carefully considered by users, device suppliers and developers, and model cells will be selected and devices will be developed. Document the verification test procedures according to the respective standards. At this time, the procedure will be peer-reviewed by experts so that they can be modified to be easily understood by anyone. In addition, user training is provided. As for the MPS project in Japan, we believe that taking these steps over time is necessary for industrial implementation aimed at regulatory acceptance for MPS. MPS has attracted much attention as a novel culture system that mimics the human body environment. In addition, it will become increasingly important as a culture tool to promote alternatives to animal testing, which are now being sought even in drug development. Through the MPS project, we continuously send out messages from Japan to promote drug development and alternatives to animal testing around the world. Last but not least, here is the summary of today's presentation and the result of the project. The MPS project in Japan is based on the needs of pharmaceutical companies or their COU, and the 4 MPS listed here are on the burn of commercialization. There are highly usable devices. There are two multi-site validation studies and 6 pharma proposed study going. And we plan to publish the result of these studies in the form of case studies. Thank you for your attention.
Okay.
Thank you, Dr. Ishida. We are running a little bit late, and I don't see for now any questions to your talk, so please post the questions Q&A, and Dr. Ishida will answer them in writing, and we can move to the next talk, I think.
Great. Thank you very much. The next talk is Megan LaFollette of the North American 3Rs Collaborative.
Awesome. Thank you for having me. Let me just get one moment to pull up my slides. Okay. Good morning, everyone. Aloha. I'm here in Hawaii on vacation, but I'm still truly excited to talk to you on behalf of the North American 3Rs Collaborative MPS initiative about a roadmap to overcoming obstacles to MPS regulatory acceptance, specifically with perspectives from technology providers. But I do want to start off the talk by acknowledging all of the members of the MPS initiative, particularly those in the regulatory subgroup. You can see all their names here with the regulatory subgroup, and particularly those that helped with this presentation in larger font size. This presentation is not just my views, it truly was a collaborative effort. So for this talk and for today's agenda, I have 3 main things that I'm going to talk about. First, Who is the North American 3Rs Collaborative, particularly our MPS initiative? Second, what do we see as the key aspects of the status quo that need to be defined for regulation? And finally, what is our roadmap for moving forward? So let's start with who we are. So the North American 3Rs Collaborative is a nonprofit whose mission is to advance science, innovation, and research animal welfare through the application of the 3Rs: refine, reduce, and replace. On this slide, and we do this through facilitating diverse collaboration. This slide shows pictures of just our board and our leadership team, and I hope you can appreciate their diversity. We have end users from pharmaceutical companies and contract research organizations. We also have biotechnology companies and suppliers. We also have government organizations, academics, other nonprofits, and even more collaborators. And in total, we collaborate with over 70 different institutions and 124 individuals across all of our initiatives. And this truly makes us a powerful force for implementing the 3Rs. We currently have 6 main initiatives focused on rodent health monitoring, refinement, translational digital biomarkers, microphysiological systems, a 3Rs certification course, and compassion fatigue. And of course, I'm not going to spend the time talking about all of our other initiatives today, but if you are interested, please just visit our website and sign up for our newsletter. Today, I'm going to, of course, be talking about our Microphysiological Systems Initiative. This initiative came about because we truly see the promise of microphysiological systems to reduce and replace animals in research. And we do see their advantages. After all, they can be human-specific, they can aid model selections for animal models, they provide us with well-needed mechanistic data, and they have the potential to save time and money. And we saw that there are 3 main stakeholders for MPS: the regulators, the end users, and the developers. And although in North America, we saw that there were places for the regulators and the end users to each have their own discussions, we really saw a gap for involving the developers and how to unite them in a non-competitive manner. And so therefore, although our group is open to all stakeholders and does involve parties from all of these stakeholders, it is focused on developers with commercially available systems. And this is important because developers truly are in a key position to guide MPS use and acceptance. After all, they are intimately familiar with both the promise and limitations of the technologies, and are constantly conducting tailored research and development on their own products and other different technologies. And so if we can involve them in the discussion, they can really benefit the entire field. So this is the logos of all our member institutions. We have 33, 26 are developers, but I hope you can appreciate the variety of different sizes. We have small, medium, and large organizations from many different types. And thus far, our initiative has 4 key efforts. First, interfacing with end users, gaining regulatory acceptance, creating a technology expo, and educating about microphysiological systems. I'm not going to talk much about the other subgroups, but I will be obviously talking about our regulatory acceptance subgroup today. And this is so important because regulatory guidance on acceptance criteria of MPS is lacking. In particular, there's a lack of requirements for technology qualifications for specific contexts of use. And we believe that it is in part because of this that microphysiological systems use is increasing slowly. Certainly, there have been gains made and implementation increasing over the years, but there could be more. And so gaining regulatory acceptance of these systems is truly critical for the field. And so that is what our group ultimately is focused on: increasing industry adoption and regulatory acceptance of microphysiological systems to reduce and replace animals where scientifically appropriate. So at this point, I hope you understand a little bit more about the North American 3Rs Collaborative and our MPS initiative. Now let's talk a bit about the status quo. And this is important because to know where to go, we must first know where we are. I'm from Colorado and I'm an avid hiker. And if I come to a fork in the path, I can't just choose a direction. I have to know where I am in order to chart the proper path. And there are 3 key areas that we've identified: looking at current MPS technologies and their context of use, as well as their current regulatory efforts. I'll talk about the first 2 first. So first of all, we are collecting data on commercially available technologies, and we've all actually created a technology hub. This hub will actually be live on July 1st. So again, please sign up for our newsletter or just email me to check it out. But it'll be really beneficial. It looks at all of our initiative members. It has a description of their companies, the top 3 publications, and currently you can sort it by tissues offered. So we know what systems are out there. Furthermore, we've also had all of our different member institutions fill out a context of use document. This includes the different organs and tissues they provide, whether they're device sale or CRO, and then which context of use they're in. And with that, I do want to take an aside to define context of use, because I myself was, not quite familiar with what these words really meant until just a couple weeks ago when my team educated me. And it's a concise definition of the manner and purpose of MPS or any technology use. And it's important because it determines how much validation or qualification is needed for a particular assay. It defines the domain, boundaries, and limitations for acceptable use as justified by the data. So in short, it's the manner, purpose, and boundaries. And it truly is essential for regulatory acceptance of microphysiological systems. After all, MPS are drug development tools, and as such, MPS could be qualified for specific contexts of use across drug development programs, therefore allowing multiple MPS or end users to use the same systems, which ultimately should increase availability of effective drugs, accelerate the drug development cycle, therefore getting therapies to patients faster, and enhance our knowledge of developed drugs. Now, I want to go over what our initiative members have for context of use. This is internal data that we've collected from the companies. The x-axis on this graph is the number of microphysiological systems, and the y-axis is the specific context of use. And I hope that you can appreciate both the diversity of contexts of use as well as the number of microphysiological systems. And these are systems that are either currently commercially available or very close to being commercially available. And so you can see there are 100 different systems that are working on the context of use of target identification and validation. And even for lesser common but greatly needed contexts of use, such as drug efficacy for immuno-oncology, there's still over 20 systems. So it's really great to see this, and it kind of gives you an idea of where the field's at right now. Okay. So now we've talked about those current MPS technologies and contexts of use. Let's discuss current regulatory efforts. So we do recognize that there are current regulatory and government efforts to advance the MPS field. These come in many forms, such as publication, research, pilot initiatives, and conferences and meetings. They're also being done by many different agencies, such as the FDA, the European Commission, and NIH. And what this means is that it's not just hype about these technologies, but true promise and interest in the field. And in fact, the FDA is actively evaluating MPS for a number of contexts of use. However, despite these efforts, definitive guidance is still lacking, particularly that which involves the developers. And so that brings me to our roadmap. Some wonder if we can modify the current standards for, say, in vitro tests for MPS systems, but we think no. This is an opportunity to create clinically relevant, built for purpose guidance that ultimately reduces the need for animal research. And we have 3 steps and an end goal to reach this. First, identify a few greatly needed contexts of use from end users that developers can commercially provide. We're going to do this through a survey asking end users about their MPS needs by context of use, urgent drug development areas, and gaps in animal models. Please stay tuned for our survey. We will be pushing it out to our pharmaceutical partners, but please do email me if you want to ensure that it lands in your inbox. And the final goal with this goal is just really to find contexts of use that are critical for all 3 stakeholders, the regulators, the developers, and the end users. The second is to write an opinion paper to guide regulators from the developer's point of view. And some features of that opinion paper is that it should be about guidance that follows the in vivo model, discussing best practices, and that MPS should be held to the same standard as animal models. I'll briefly go through the importance of these different parts. So first of all, we should have guidance instead of guidelines, because guidance allows MPS systems to have validity, but also movement. And it doesn't artificially advantage any particular system, but allows instead support for whatever system is scientifically appropriate to answer the question at hand. We also recommend following the in vivo model, in part because we truly believe that MPS are much closer to animal models versus the in vitro model. Again, with that flexibility, having these best practices will allow developers to justify their actions and create quality, scientifically supported systems. And finally, they should not be held to a higher standard than animal models. Just because we've used animal models traditionally does not inherently mean that they are better than MPS. And so they should be held at those same standards. So the third step will be to produce consensus-based guidance for regulatory acceptance involving all 3 stakeholders. And finally, the end goal is to create an ICH working group to define the testing criteria. So just to summarize our roadmap, first, to identify critical contexts of use that are applicable and good for all 3 stakeholders. Second, to write an opinion paper for regulators based on the developer's point of view. Third, produce consensus-based guidance. And finally, to create an ICH working group to define the testing criteria. Ultimately, our aim is to create global, harmonized guidance for microphysiological systems to make this technology as regulatory accepted as in vivo models. And with that, I'd just like to thank all of our current institutional sponsors for the North American 3Rs Collaborative. We are a nonprofit and couldn't do it without their support. And once again, wholeheartedly thank our MPS initiative members. This really is a collaborative effort, and they've put so much work and do so much exciting and impactful work with their group. And with that, I'd just like to again encourage people to join na3rc.org to learn more. You'll find out about our MPS initiative if you are interested in joining us. As I said, we are primarily focused on developers, but we are open to all stakeholders. You can find some more information there, and you can email me directly at meglafollett@na3rc.org. And with that, I'll take any questions.
Thank you, Megan. That was really nice overview of your ongoing work and plans and roadmap. I have a question. do you have any timeline in mind how the roadmap will be followed step by step, steps you presented?
Yeah, so the survey is in development now. I think we want to get it out in maybe the next 4-ish weeks. And I know the plan, and actually we are writing 2 white papers right now, which I think should be done by the end of the year. I'll have to double check the charters, but I'm giving estimates right now. I believe the plan is to at least be having a proposal to the ICH, I believe by December 2021. But again, I'm not 100% sure. And if the person who asked is interested, I can double check the charter, but we did have each of the subgroups outline their goals, the timeline, and exactly when they plan to do it. But yeah, those white papers are already in development. They're moving ahead pretty well.
And I have another question. So I saw FDA on one of the logos, but I didn't see any other regulators. So how, and I understand you are heavily loaded with pharmaceutical companies. How close is the relationship with regulators? How are they involved? From which step are they in your consortium?
So they're involved in each of the subgroups. And then I know we, even after today, we have planned to reach out potentially to some other regulators. But I'm not sure I completely understand about like how involved are they?
In the discussion and in the development of the roadmap.
Okay, yes. Yeah, I mean, so we try to involve them at every step of the way as much as possible.
Okay. So there is one, not question, a comment on the chat, on a Q&A, which I will read, which just echoes what you just said. Many in vivo models, even the ones used in OECD, that the test guidelines were never formally validated. MPS have the possibility to be much better than in vivo models. We should not compare in vitro methods with animal data, but develop human relevant methods. Thank you.
Yeah.
The comment came from Andras Stuckey.
Yes.
And I don't see any further questions popping up. Please check Q&A. If any comes, answer them, and then we can move with that to the next speaker, our last short talk speaker. Thank you, Megan.
Thank you.
Okay, thanks, Megan. Thanks, Lena. We are moving on to our final speaker for the short talks, and we have Rob Vanderbrew of the National Institute of Public Health and the Environment.
Yeah, can you see my screen?
Yes, you're good to start. Thank you.
Okay, thanks very much. I would like to thank the organizers for giving me the opportunity to present our study. Next slide, please. In this talk, I would like to discuss the opportunities and issues related to organ-on-chip application in the safety assessment of chemicals and safety and efficacy assessment of pharmaceuticals, and the steps needed to achieve this goal. Next slide, please. For the transition to a more predictive and 3R compliant drug development and regulatory safety assessment, there are basically 2 scenarios. The first one is an evolutionary approach that's based on the current practice of replacing one animal test at a time, replacing by a set of non-animal methods that together predict the endpoint for which the animal test was designed. It is carried out stepwise as standardized and validated non-animal methods become available. And the relevance and predictivity is defined by comparison to animal data. There are already some endpoints in use: irritation, corrosion, sensitization, and genotoxicity, and they are in guidelines from OECD and ICH. But importantly, there's a lack of accepted non-animal methods for the more complex endpoints like acute toxicity, repeat dose, carcinogenicity, reproductive toxicity, and neurotoxicity. Next slide. The alternative scenario is a revolutionary approach. And that is based on the identification of crucial mechanisms that are relevant for human disease. It comes down to mapping critical rate-limiting steps in physiological pathways that lead to toxicity by chemical perturbation. They're tightly linked to adverse outcome pathways, and the coverage of key events in those adverse outcome pathway determine the relevance of the testing strategies. Relevance in this sense can be defined as a measure of the extent the method is covering events of a molecular network that mediates toxicity, and the predictivity, the ability to determine whether a chemical can perturb this network to the extent that it leads to irreversible adverse effects. Next slide, please. Well, then the question arises, how do organs on chip fit in? Well, the main advantage, as we look at it, of organ on a chip technology is the possibility to more closely mimic in vivo processes compared to the usual 2D in vitro models, and thereby increase the biological relevance of both the pathways and the endpoints. Organ on chip will usually contain a larger biological domain, and therefore it covers a larger segment of AOP networks, and thereby it facilitates studying the connection between key events that are rather remotely related in a network. Of course, organ on a chip has some downsides. It's still relatively low throughput. Compared to suspension cultures, there's the complexity of the kinetics and the difficulty of interlaboratory standardization that, of course, has to do a lot with validation. Next slide, please. The applicability for organ on a chip for the chemical and pharmaceutical safety evaluation, there are basically two starting points. The first one is the tissue-based, and in that sense, the organ on a chip is valued in the sense that it can recapitulate the characteristics of the tissue, and the qualification of the organ on chip is based on a series of chemicals or pharmaceuticals, with some of them requiring metabolism. And the effects are monitored at the tissue level, and the evaluation is whether it agrees with in vivo rodent data or human data. Next slide. The other starting point is mechanism-based, and it is based on AOPs. And the organ on chip is interrogated, is the molecular initiating event operative, which key events are operative, and of course, more downstream key events and the adverse outcome are less likely to be operative, because especially for adverse outcomes, a more complex system, a complete organ, or even a complete organism is often requires. But it provides an in-depth understanding of the extent to which the AOP is captured in an organ on chip. Next slide, please. Well, one slide on the reference compounds. One may identify several groups of reference compounds. The first group delineates the chemical and biological applicability domain, for instance, chemical classes, mechanisms or mode of action, and molecular initiating events and key events. The second group may be identified that evaluates the transferability of the organ on a chip model. The third that relates the outcome of the organ on chip model to known clinical events, of course, preferably in humans, but also in experimental animals. And the fourth group of compounds is that The ones that require special attention, for instance, the one that require metabolism, or so-called false positives that have an adverse outcome at a relatively low dose that is seen in animals but not in humans. And of course, the rather low throughput of organ on chip limits the number of compounds to be tested. Next slide, please. For standardization, there's of course a rigorous and strict standardization of organ on chip methods necessary. And I would like to mention two helpful guidance documents for this. The guidance documents from the OECD 211 and 286. Next slide, please. Well, regarding the interlaboratory transferability of organ on chips, in our opinion, one should not aim for 100% copy-paste of organ on chip between one laboratory to another, because there are differences in infrastructure, equipment, culture medium, and especially serum batches and sources of biological material. The essentials of the standardization should be qualified, look at the positive and the negative controls, and score the test compounds relative to these controls. Next slide. So standardization. Oh, sorry. This one. Standardization really is a balance between the progress and the precipitation in standard organ on chip models. The organ on chip models, both regarding the engineering part and the biology part, should be transparent, and they should adapt to the rules of friend, fair, reasonable, and non-discriminatory principles. Next slide. Regarding regulation, regulatory scientists should keep pace with the advances in the basic and applied science and technologies, while at the same time critically appraising new technologies for fitness of purpose and use. As mentioned earlier, due to the early stage of MPS technologies, specific guidance on safety assessment by MPS models is not yet established, although initial steps are taken by the pharmaceutical industry. Next slide. Until now, I talked a lot about the perspective from the chemicals, and now more from the pharmaceutical industry, because their division is a bit different. There's no need for standardized guidelines, since many non-animal models are developed in-house, and their variability cannot be captured in a guideline. And here the term qualification can be coined. For pharmacological characterization, there's purposefully no or very limited guidance, as this would lock innovation. And also it is expected that innovation will go too fast for standardization of test guidelines. The applicability of organ on chip system is most viable in the drug discovery phase or the early development phase. Next slide. Regarding the 3 R's in pharmaceutical industry, well, there's a staggered product development, and organ on chips may be used to defer in vivo testing to a later development stage. And by attrition, this may result in less animal testing. Next, organ on a chip can supplement or replace pharmacological mode of action and proof of concept studies, and thereby identify relevant pharmacodynamic and toxicity markers to monitor in animal toxicity studies. and this may result in fewer animals. Also, organ-on-chips may provide relevant monitoring markers in clinical studies, and as a mention of care, even if you have a predictive organ-on-chip model, this does not always lead to a reduction in experimental animals. It can also lead to another layer of knowledge on safety or reduction of the attrition rate in drug discovery. Next slide. So to come to the conclusions, thorough and consistent validation is not reached yet. Further work is needed to reach broad acceptance both by pharmaceutical companies and regulatory agencies. Extensive characterization and method description, high quality SOPs and a data repository are required. The data repository on effects of applicable molecular initiating events and key events, important to underline the specifications of the organ-on-chip. The technology underlining the organ-on-chip should be open source. Biology, engineering, and software should be standardized, and developers should interact with regulators already from the start of the organ-on-chip development. Next slide. Yes, I would like to mention the UROC project. This is the EU-funded training network project, where PhD students each develop their own organ-on-chip model. But the UROC project also has a validation board consisting of experts from industry and regulatory bodies. And the PhD students are trained in validation requirements as a basis for their future career. In fact, the UROC project initiated the study that I presented today, and the publication is submitted. Also, I would like to mention, as has done before, European Organ-on-Chip Society that is really building bridges across different parts of the organ-on-chip network. And my final slide, next slide, is I want to acknowledge Marlon Schneider, Michael Oelgeschleger, and Tanja Burgdorf from BFR in Berlin, Peter van Meer and Peter Teunissen from the Dutch Medicines Evaluation Board, and my direct colleagues Anne Kienhuis and Albert Piersma from the Dutch National Institute of Public Health, and of course the UROC project. Thank you very much.
Thank you, thank you. Very nice talk. I have a couple of questions to Europe. First is from Donna Mendick. Identifying positive and negative compounds for testing is always difficult. How would you identify such compounds, and would you have a large group helping to make these decisions?
Yeah, it is indeed difficult to define them. It's not something that you look up in a book. You have to be aware of the mechanisms involved. That's important, and often they are defined by a group of experts, so by consensus together with regulators.
And there is another question from the Q&A from anonymous attendee, also regarding reference compounds. Whether you could give an example. In Vivo, for example, they use estradiol in toxicity study for endocrine disruption. If you use toxicity study with drugs or chemicals using MPS, what should we use? This is the question.
Oh, can you please repeat it?
So the attendee is wondering about the example of reference compounds. For example, estradiol for endocrine disruption in vivo, which also used in vitro. Actually, what he's using, whether you can give some examples on reference compounds used in MPS.
Well, I can give examples of reference compounds, but not in this specific area because it's not my area of expertise. Sorry.
Okay, so maybe you can type an answer if you like later.
Yes.
Another question is about standardization, whether it means in regards with the source of cells used to generate MPS. Source could be very diverse, and if from patients, the notion of normal tissue is questionable. So how would you use the standardization in terms of the cell sources going into the MPS?
Well, so far, we've used only from, say, ATCC cell lines. That's step one. And I think that after that, you have to make a one-to-one comparison with first material from normal human donors or from stem cells, and then from patient-derived stem cells. I think it's, it should be done in several steps with more and more complexity.
Okay, and there are two questions, actually one comment and one question which comes hand by hand. So Cliff Roper has been talking about the validation of in vivo tests. We have to keep in mind that only one in vivo test, which is LLNA assay, was formally validated. And what about standardization? Interlaboratory standardization of in vitro is important, but what about the interlaboratory standardization of in vivo? We should keep in mind that we need to have the same standards for in vitro and in vivo. And following that, there is a question from Tamara Zidek, how to deal with the problem of species-specific differences when comparing human MPS data to animal data. Is the light of high clinical failure rate in drug development, would such an approach really make sense?
The best answer would be to also use cells from the animal in an MPS system, so you can see whether if you find differences, whether it is caused by species difference or not. On the other hand, when you, from the mechanistic point of view, it should be, you should not need animals to find your answer.
Yeah, and would you like to comment on Cliff about standards for in vitro and in vivo? So it looks like we have higher standards for in vitro to reach, which were never put.
Well, in my experience, the more complicated the animal assay becomes, the more differences in outcome you get. So maybe, and that is, in my view, one of the big problems with animal studies, that once you embark on a disease model using animals, the variability in the results becomes higher. And for instance, in arthritis models, in animals, it can go in various directions. So in that sense, to me, that's one example where I think MPS is very promising.
Okay, thank you very much. We answered all the questions in Q&A session, which are there so far, and we can move to the panel discussion.
Okay, thank you so much, Rob, and thank you, Lena. For our panel discussion, we have Dan Tagle of the National Center for Advancing Translational Sciences, who will moderate a discussion with with international regulators on perspectives on regulatory acceptance. The panelists include Anne Gourmelon of the OECD in France, Sonia Becken of the European Medicines Agency in Belgium, Seiichi Ishida of the National Institute of Health Sciences in Japan, Jean-Loup Dorn of the European Food Safety Authority in Italy, Donna Mendrick of the Food and Drug Administration in the US, and Elizabeth Menden of the Environmental Protection Agency in the US. Please do write any questions that arise during the discussion into the Q&A box, because after the panel discussion with Dan and the panelists, we'll have an opportunity to answer your questions, and Ricardo Barile of University of Cincinnati will moderate a Q&A with the questions that you put in the Q&A box for the panelists. I will ask Dan and all the panelists to unmute yourself and turn on your cameras. And Dan, you have the floor.
Well, thank you so much, Elizabeth, and a good day to everyone. And thank you for hanging in there for this last session, which is the panel discussion. And hopefully this will be a very productive discussion of how we could move microphysiological systems to regulatory acceptance. So certainly this is a very distinguished panel. And so why don't we start this session by having each of the panelists, and I'll tell you which one to go first, go and introduce yourself briefly about your organization, and perhaps a little word or two about what your organization is doing in terms of moving microphysiological systems towards regulatory acceptance, either having submitted a package for regulatory approval or actually as a recipient of such a submission. So why don't we start with Anne? If you can introduce yourself and say something about your organization.
Thank you, Dan, and good afternoon, good morning, or good evening, everyone. I'm Anne Bourmelon. I am a principal administrator of the test guidelines program at the OECD. So I presume most of you know what this program is about. In the context of this event today, I would maybe mention that most of our work program at the OECD today is dedicated to the development of non-animal methods. And we are very much versed into developing, of course, methods and combination of methods as well that can be used together to predict adverse outcomes. So together with member countries and industry partners and stakeholders, we invest a lot of our resources at the OECD in programs and activities and projects that will pave the way for the future for maybe MPS to become part of the, of the landscape of standards that regulators will be able to use data from. And just to name a few of the activities, there is, of course, a major activity on the development of adverse outcome pathways at the OECD, contributed by member countries and many research groups around countries. Also we are developing a lot of frameworks to enable the emergence and the regulatory use and ultimately the acceptance of non-animal methods and new approach methodologies, such as integrated approaches to testing and assessment. We have developed some frameworks and we developed a number of guidance documents that were mentioned in the course of some of the presentations previously. So yeah, just I'd like to just stop there and give the floor to others.
All right, thank you, Anne. Sonia?
Yes, hi, good morning, afternoon, evening, everyone. My name is Sonia Becken. I am currently working at the Belgian Medicines Agency as a coordinator of the team of non-clinical reviewers. However, I'm also member of the EMA, the European Medicines Agency's Safety Working Party, and have been chair of its 3Rs working group. So in that sense, it's clear that the European Medicines Agency and all the national European agencies are clearly committed to the 3Rs. Within the EMA's regulatory science strategy, it's clearly mentioned that there is a specific recommendation dedicated to the leverage of 3R methods and their qualification. Within its 3Rs group at the level of the EMA, we have a lot of activities and discussions also related to 3R methods, of which MPS technologies are, of course, an important part. And the different experts that are present within the working groups of EMA are also highly involved in all the European and international initiatives that are ongoing with regards to discussions on qualifications of MPS technologies, such as the European Society for Organ on Chips, for instance. We have had a workshop already in 2017 on MPS technologies, which brought together all the stakeholders at the level of the EMA, and we had a discussion really on practical aspects of how to move the technologies forward and how to come to methods that could be ready to investigate for regulatory acceptance, which of course encompasses all the topics that we have heard today related to qualification, reference compounds, and context of use. So in that sense, we are heavily committed to help moving this technology forward and liaise also with our colleagues at the level, certainly of FDA, as well, to see whether we can join forces in that sense. Thanks.
Thank you, Sonia. Seiichi, you're next. We heard from you earlier, but can you tell us a little bit more about what AMED and what the efforts are in Japan?
Yeah, so my name is Seiichi Shida, and I presented my slide as for the introduction of Japanese situation in the MPS development. So I'm a guest researcher in National Institute of Health Sciences, and we are and I'm participating in the AMED MPS project, which is one of the leading MPS projects in Japan. And at least now, in Japan, MPS development is MPS is now wide acceptance of MPS technology in pharmaceutical industry is in the development status. So we are talking each other. We means the pharmaceutical companies and suppliers and the regulatory side. And National Institute of Health Sciences are doing some experiment with MPS for the regulatory acceptance, but it's still a preliminary stage. And now the PMDA, Japanese Pharmaceutical and Medical Devices Agency, start to join the our project. So in Japan, it's just the beginning of the time of the talk between the regulatory side and the developer side. Yes.
Okay, thank you, Seiichi. So next on my list is Jean Lu. You're muted.
Yes.
Thank you.
Hello, everyone. Thank you very much for inviting me to participate to this discussion panel. So I'm Jean Loudon. I work at the European Food Safety Authority for the Scientific Committee and Emerging Risks Unit. We develop guidance documents, and we also focus, I'd like to mirror what Sonia and Anne said, we focus a lot on new NAMs, new approach methodologies, particularly in the area of physiologically based modeling and the link with in vitro assays. So I take it that the MPS is very relevant to these developments. And we have a current research project looking at nano risk assessment, comparing in vitro methods and MPS. Thank you.
All right, thank you. Next, Donna.
Hi, I'm Donna Mendrick. I'm the Associate Director of Regulatory Activities at NCTR, which is a research center of the FDA. I'm also, as you heard today from Susie's presentation, I'm co-chair of the Alternative Methods Working Group. I am not a regulator, but I have friends who are, and I've talked to them about some of the questions that Dan posed ahead of time. I'll do my best to answer them. Thanks, Dan.
Thank you, Donna. And last but not least will be Elizabeth Mendez. You're muted, Elizabeth.
Yes. Hi, my name is Elizabeth Mendez. I'm the Senior Science Advisor for the Human Health Effects Division in the Office of Pesticide Programs of the US EPA. We have been an organization that has been at the forefront in our agency in developing and using new alternative methods. So this symposium or workshop on MPS is definitely of keen interest for us. And I'm grateful for the opportunity to participate in this panel.
Thank you, Elizabeth. Yeah, so let's start off the discussion by, I think you were shared a set of questions that was developed by the organizers. So in the sense that there is certainly a lot of interest in this growing field about where the next steps would be. And I think the next logical step would be certainly regulatory acceptance and industry adoption. So from your various, where you're sitting in your various agencies, have you seen or become aware of any submission packages, either through IND or regulatory submission, or have you been involved in any of them? And if not, what do you think would need to happen? I think we've heard some challenges throughout the morning in terms of what it takes to get regulatory acceptance. So in your mind, what would be the biggest hurdle that the developers and the users need to implement in order to get to that point?
Dan.
Yeah.
So CEDAR has not seen any INDs with microphysiological system data. We keep hearing from companies that we will be seeing it, so I'm assuming it's a matter of time, because obviously the things churn through slowly at a pharmaceutical company as we get the data together. So I think, and the FDA's certainly been, a lot of people have been trained in the FDA, they're aware of MPS and what they can do. So I think that's a matter of time. And I think in a lot of cases where there's new technology, the sponsors are very nervous to submit data. Mm-hmm. And that's why we always encourage people to come and talk with the FDA early, because I don't think the FDA regulators are that scary, but people are just very concerned. So I think that eventually we'll see data. I doubt it's going to be for pivotal studies, but I think it's going to be for add-on studies and things will complement, and that will start making everyone feel comfortable submitting such data.
Okay, thank you, Donna. I'll have a few follow-up on that one. Sonia.
Yeah, and maybe I can give a kind of parallel answer as Donna. At the level of EMA and at the level of the national agencies, we haven't seen a submission of MPS data for supporting clinical trial applications or for marketing authorization applications. I must say that we have given the message a lot already that we are really open and keen on seeing those data on our tables, because it's the only way we will get to know also the systems is to look at the data that are being generated by them. But there is kind of a hurdle indeed to submit the results obtained in the MPS systems to us. There are specific mechanisms available to submit data, as we have a mechanism called the Innovative Task Force, where method developers can come directly to the EMA and discuss in a free of cost procedure possible steps towards qualification. And really it's a kind of stepwise approach where method developers can be accompanied to grow towards qualification. But even also in that procedure, we have not seen submissions of MPS systems yet, although we are, as I said, very enthusiastic to see the systems and to learn and grow together.
Mm-hmm.
Thanks.
Well, there you heard it from two major regulatory agencies that they're willing and eager to hear. Yeah, Elizabeth.
Yes. So if I may jump here, we have received one submission in regards to microphysiological systems relating to lung toxicity, and that was used in concord in tandem with a computational fluid dynamic model. And in that instance, it was used because the architecture of the respiratory tract in a rodent obviously is very different to the human, and we were getting some results that we felt were probably not human relevant. It was the submission was the subject of a science advisory panel meeting of the Federal Insecticide, Fungicide, and Rodenticide Act. And we received very good feedback from that panel, and we anticipate that we will be using that one submission will be driving the inhalation risk assessment for one of our chemicals.
That's fantastic. Thank you, Elizabeth. Anyone else, either part of a submission package or have received them? So if not, then let me go back to I guess for Elizabeth, Sonia, and Donna. So you mentioned that you may have heard, or in the case of Elizabeth, actually have a submission, and it seems to be not so much as a standalone in vitro model MPS data only, but in this case, supplemental to modeling. Do you anticipate that to be the case for any of the submissions coming forward in the future, or would any of your agencies be receptive to just MPS in vitro data submission?
So I'll tackle that one first, I guess. So I guess as we become more familiar with the technologies and what they can do, I anticipate that eventually we'll get to the point where we may be able to rely on MPS data exclusively. But at this point in time, I think it's going to be in conjunction with other data sets, and also depending what the fit for purpose is and what our purpose are. We can use these data in a variety of ways. It could be for point of departure derivation, but it can also be to help us further tailor additional in vivo testing that we may need. So it all depends on what the purpose is that we will be using the data for.
Yeah, go ahead, Sonia.
I really would like to reiterate, in fact, what Susie said and a number of other speakers said. It's the importance of the context of use. There are so many different models that are being developed, being test-driven in different companies and method developer sites, and they have all a particular context of use for within that organization. And we haven't had a real dialogue one-to-one with the regulatory authorities to define for one method what would be its context of use, and then it can be very variable. We had a very interesting workshop at the level of the UROCs regarding different possible contexts of use of these systems, and it's extremely broad when you look at the drug development pathway. It goes from really efficacy testing, proof of concept testing, mechanistic support for investigating translatability to humans and so on, to safety testing and personalized medicine applications. And each of these specific contexts of use will require different qualification standards, as has also been mentioned in the presentations today. And I think that that is really important. It's difficult to say now what will be the overall use. I think it will be really dependent upon the model, and we will have to grow in that, and confidence will be built in a stepwise approach.
Thank you. So I have a comment from Jay Hickman indicating Hesperos, his company, has submitted a package to CBER in collaboration with Sanofi for CIDP. So I guess we're going to be eager to hear the outcome of that one as well. So here's a question perhaps to Donna. Said the ISSO 10993-23 was a success to which many industrial partners contributed. However, we do not see it yet being considered by the FDA. This is rather sad after so many years of efforts of various groups working on the subject. Would you care to comment on that one?
I actually don't know anything about it, so I can't comment. Sorry.
Okay. All right. Okay, and then for the audience, Clive Roper shared a link to what Elizabeth had just mentioned about the study that she just described. So here's an interesting question from Martin Stevens. Said MPS are a prime example of new alternative methods or new approach methods, a terminology that is gaining traction, especially in the US and Europe. All parties are excited about the potential of this new methodology to limit animal use in the long run. However, in the short term, some of the alternative community are concerned that the new alternative method or new approach methods terminology would obscure the historical language of the 3 R's, especially the replacement. Is there anything else to be said in this webinar to address this concern? So are we truly in a position, I guess, to, with this methodology, go to the point of replacement? And if so, what are your thoughts on that?
If I may?
This is Donna. I think that's exactly the issue. I don't think we're ready for replacement. And again, the alternative methods include a lot of different things. So things don't actually have to replace animals. Unfortunately, they can add a lot of value without replacing animals. Mm-hmm. So I think that's why this terminology has come along. And we don't like the term non-animal methods because, in fact, again, sometimes you might need to use animals, or maybe you can use them in a better way than what's used now.
Yeah, that's a great point, Donna. Jean-Luc?
Yeah, thank you. Yeah, I'd like to come back to what Elizabeth said. So in the context of food safety, we of course are trying to derive points of departure, or so-called here reference points for chemicals. One of the hurdles to replace animals is the fact that the legislation requests, of course, all these animal studies. So in the context of the food safety area, I would say that we need to put quite a lot of efforts to actually demonstrate how to use these MPS models combined with other types of information, such as in vitro metabolism studies, benchmark those derived on in vitro systems and so on, and compare the outcome with the in vivo data set so that we may be able to move forward to replace the animal testing. That would be my take on it. Lots of case studies investing different types of compounds, persistent, non-persistent, and really demonstrate how we can use these tools.
Mm-hmm. All right, thank you. You have your hand raised, so go ahead and jump in.
Yeah, thanks. Yeah, in relation to this question, some experience from the OECD I can share is that we have seen that the emergence of new alternative, new approach methodologies was better accepted if they were decoupled from the question of the issue of replacement of animal methods, because otherwise there was a tension of the regulator having to accept something in exchange having to give up something else. So for the acceptance, the adoption of emerging techniques, it was better to completely separate these two discussions. And I think the discussion of the replacement is a fair one to have, but maybe separately. And rather than completely talking about replacement, and deletion of or cancellation of animal tests. What we've been hearing recently is rather to restrict little by little the existing animal methods, providing guidance on what is less preferred methods, and so expressing an order of preference and putting the animal methods, because there will always be certain utility for an animal method, and it's probably better to address that question separately and in a specific context of use, because they deserve their own attention, these animal methods, and what they might be useful for and what they shouldn't be used anymore for.
Right. Thank you, Anne. Certainly it's replacement is a laudatory goal, but it should be kept separate from the context of use, and ultimately we will get there, but that's not the near term. Elizabeth, you had your hand up.
Yes, actually, you just said what I was about to say is the ultimate goal is indeed replacement, but at this point in time, I think that this field is still too novel for regulators to really move in that direction yet. I think that as we gain more confidence, as we see more of these data sets, and we can see what processes it does or does not recapitulate, then we can start thinking about a replacement. But as Susie said at the very beginning of the summit, all of these changes take time, and it's going to take time and experience for the widespread acceptance of these new technologies.
Thank you. Sonia, you had something else to contribute as well?
Yeah, in the whole discussion, indeed, we are very committed to the 3 Rs. Certainly here in Europe, it's enshrined in our European directive. But we also have to consider that we are looking at all these novel approach methodologies in the context of having better tools that give us a better idea about human efficacy and human safety with regards to drugs. And that's also what we really need to aim for, to have better predictive tools. And if this is without animals, this is good thing, but we want better tools. And that's also why we are looking at all these MPS models. Thanks.
Mm-hmm. Thank you. So I have a question from Frederick Sanajas. They said, can we imagine that MPS experimental study data will be first part of the investigator brochure instead of highly regulated INDs? It might be an initial proof of interest and easier acceptance by diverse regulatory authorities, the FDA, the EMA, the PMDA, to confirm a proof of concept, efficacy, or safety risk assessment. Thoughts on MPS data being part of investigator brochures? And how is that different, let's say, from creating some sort of a safe harbor environment? To open up discussions within users and regulators.
Okay, then can I give a first shot at this?
Go ahead.
Okay. Of course, we would love to see the data as like proof of concept information generated on MPS. We are looking at proof of concept data coming from various in vitro models in investigators' brochures. So if this is coming from an MPS model that has been well qualified for that specific context of use, very welcome to submit that in an investigator brochure. If it's also to indicate ideas on adversity or on translation of animal findings to humans, also that could be very helpful. How is this different from a safe harbor procedure? A safe harbor procedure would have to be discussed in collaboration with the respective regulatory authorities, whether this is possible. And then this is more in a path going towards qualification of the methodology where data is gathered in a real life situation without it being considered in benefit risk assessment for a specific product. So in that sense, this is a kind of different trajectory than submitting data on proof of concept, which is always possible within a specific dossier.
Mm-hmm. Is that only with EMA, or is that true for other agencies as well?
May I?
Yes, Jean-Luc.
Yeah, so well, I think again, as I said, we have dossier packages which are framed by legislation. However, an opportunity for proof of concept would be to look at certain contaminants like undesirable substances in food. We can think of mycotoxins and various other chemicals. So these could be investigated using MPS. And again, the outcome could be compared with the historical risk assessments. So there's probably an option there for the food safety area.
Okay. Anyone else? Okay, here's a submitted question. MPS and organoid, which you think will have a faster acceptance for in vitro model?
That's a difficult question.
Donna, I don't think it matters. It depends on the context of use and how well your assay does.
Yeah, and given that organoids and MPS or organs on chip have actually, in some cases, have actually been combined in the sense that I guess it's something called synergistic engineering, that some organoids are actually placed on microfluidic devices. So at some point, there is a convergence of technologies and things. Right. Did you have anything else to add, Donna? You're muted.
Oh, sorry. No, I have nothing else to add. Thanks.
Okay. Here's a comment from Stefano to support Sonia, what Sonia was saying. EMA is interested in discussing with developers new alternative methods and the pathway towards regulatory acceptance. So he provided a link to the task force, the innovation task force that Emma have put together, and that's on the chat. So just directing your attention to that. Okay, here's where you put in your crystal balls and look into it. So somebody posted a question in terms of timeline. It does take time, but how much time do you think it will take to, I'm not sure what the question is, whether it's replacement or actually more, but let's tackle both. How much time does it take to, because we're starting to hear submissions coming in or potentially coming in. So I think that's probably something that will inevitably will be coming anyway. So I think this question is more about replacement, I would think.
So this is Donna. Unfortunately, I think it's going to take a long time. First of all, it takes a long time for communities to accept a new approach. This can be a biomarker in the clinics. I mean, people unfortunately are resistant to change. Everybody is. We're hardwired that way. So, you have to get people comfortable with a new approach, regardless of whatever it is. I mean, I think that's the first part. So yes, I think it's going to take a long time. And to start to replace an animal, again, Brian Berridge talks about when you do a classical toxicology study, you look at 55 organs. Are you really going to replicate, when can you replicate all of that in vitro? Because you're going to need to do that if you want to replace a classic animal toxicology study. So it is going to take a long time. We're not there scientifically. And then again, you have to get people's comfort level built as you move along as well.
Okay. Elizabeth?
Yeah, I think it is going to take a while, but I think as I mentioned earlier, we at the EPA have already used one instance of an MPS for one of our risk assessments. So it all depends on, again, the purpose and the fit for purpose. And what systems we're looking at. In our case, we were looking at a very defined type of toxicity. So it was a little bit easier for us, rather than having to look at, as Donna was saying, at 55 organs. We were concentrating on one organ system that allowed us to move a little bit faster. But even in that circumstance, it took about, I think it was 2 to 3 years from the moment the submission came in our doors to the moment where we were ready to accept it and use it for regulatory purposes. So I don't see us moving into replacement within the next 5 years or so, except for us mattering here and there.
Mm-hmm. Thank you. Sonia, anything to add?
Yeah, I really would like to support also what Elizabeth said. It's really dependent upon the purpose. What is the context of use again, but also what is understood under regulatory acceptance. Is it to submit data generated in an MPS to support a specific development for product, to explain a mechanism of action or to explain a mechanism of toxicity within one particular dossier? That can be regulatory acceptance. Or is it really to target a change in a guideline requirement, or to defer certain non-clinical studies to a later stage, or waive them maybe? At that point, of course, you are looking at a much longer time scale than in the first condition. And what kind of time scales are we talking about? If we look at the process of revising ICH guidelines, we are looking at several years to bring everybody in the same alignment around the table. And 5 years is indeed not an underestimation in that context. But we need to be progressive and think about how we will be able to place these methodologies in the current testing paradigm. And that will even take more time, because that's like Rob said, it's a revolution rather than an evolution.
Yeah, well, certainly, thank you, Sonia. But certainly we've seen quite a remarkable revolution that in biomedicine and biomedical research happen in the last year or so during this pandemic, when we saw academic institutions, government, private, working together to come up with vaccines and diagnostics. Is that something that there are going to be lessons learned that we could all harness together as end users, stakeholders, funding agencies, as essentially as a community of investigators? What are the lessons learned from what we have seen transpire in biomedical research as it relates to the pandemic, in terms of how we could focus that type of lessons in moving this technology forward? Any particular thoughts or insights on that? Does it take international harmonization, international community working together towards standards? What are the biggest things that we can and should tackle as a community of scientists?
So I think that one of the things.
Go ahead, Elizabeth. You had your hand up.
Yeah. So I think one of the things that we need to keep in mind as it pertains to SARS-CoV-2 is that although we have seen a historic effort to develop vaccines in a time frame of 10 months, the reality of the matter is that the RNA technology had been slowly progressing for many years prior to that. So it wasn't a novel technology per se, it was the application of it to a public health emergency. So it's a little bit different. The technology was already fairly well developed when SARS-CoV-2 came into the forefront, but that's not quite the same with some of the other MPS systems that we're talking about today.
Okay. Sonia?
Yeah, I think it's not really magic. I think collaboration and communication, as Susie said, it's really the key. And we need to work together. And there are already a lot of big initiatives ongoing to work together. And we need to join forces, and regulators, as said before, need to be involved really from the beginning. Maybe in the past initiatives for other technologies, regulators have been involved maybe a bit late, where the context of use was less well defined. And I think here we have an opportunity to really involve all stakeholders at the beginning and to target the efforts, what makes the probability of success in certain contexts certain. But we need to join efforts. And if I mean join, it's really a global effort that needs to be done to move these technologies forward.
Okay,
Thanks.
thank you. Anne?
Yes, yeah, you were wondering whether standards and harmonization is needed, and I think from the experience we've seen, I guess you know, success stories, case studies need to come first to have a critical mass of evidence to convince regulators that maybe in certain areas it's time for a change, it's time for a new standard, but putting the standardization first as a need. If there is not sufficient experience, it could lead to failure as well. So we believe very much at the OECD in case studies, and we encourage that to really bring the regulators on board and let them have the experience. And yeah, standardization and harmonization can come next when there is more certainty, more confidence in the new technology or the new approach.
All right, thank you. So we have a question from Clive Roper. He said, when we run animal tests, we have no concern about adding in new endpoints. In fact, we do this all the time. Add two study types together to reduce animal use and new endpoints to generate more data. And it is not questioned in the way we question non-animal models. I would not want to see the same debate with adding additional endpoints with animal tests, as we must increase the data from the animals. What does the panel consider with new in vivo endpoints? Do they ask those developers the same questions? Is there a framework for these developers? Jean-Loup, you had your hand up.
Well, no, I just wanted to reinforce what Anne said. Case studies, I think, are definitely important. Structuring the data that comes out of those technologies, again, maybe OECD harmonized templates would be a good way to actually structure this data set. And of course, later on, probably guidance documents on the broad use of NAMs under different regulatory contexts could be quite a good way to actually move this forward. But again, we need the material to the proof of concept for different regulatory contexts.
Mm-hmm. Thank you.
Thank you.
Any other thoughts on additional endpoints? Okay, if not, question from Barb Jones. Amy Avila at FDA published a paper on NAMs and suggested that we should seek to utilize NAMs in disease models where an adequate animal model does not exist. Are you working on specific guidance on what criteria you will use for adequate or for inadequate animal models? I think this is also in line with Jay Hickman's question about focusing some discussion on models of efficacy.
So actually, the FDA has a guidance on these types of models. It was published, I think, in 2014. It's the animal rule. And this paper was not only about disease models, but also actually listed a number of data gaps where NAMs would be helpful, and also lists so it lists the types of testing we need answers from for drugs, and then listed the data gaps and where NAMs can fit. But again, as far as the animal models, I would tell you to look at the guidance from FDA called Product Development Under the Animal Rule.
Mm-hmm. Great. Jean-Luc?
In EFSA, we've had quite a few cases where, especially for contaminants, where we have the opportunity to test whether the rat or the rabbit or the test species are relevant to humans, because we have enough data to do that. So we have the epidemiological data in humans, and we can do mode of action analysis to see whether that the rat would be relevant. And we have a guidance document on how to look at the analysis of biological relevance. So yeah, again, yeah, there are some elements. Unfortunately, again, legislation may restrict, the submission of dossier data using a standard test species. So that's going to be an important aspect to tackle as well, is to think through these default species and maybe inform on the molecular aspect of what we learn from those default species compared to humans, because I think that could also help.
Okay. Well, thank you. We're at the end of our panel discussion, and certainly I would like to thank the panelists for your expertise and for your time, and certainly for your excellent answers. Is there any particular question that the organizers want to ask the panel before I turn it over? Elizabeth Baker or Camilla, any particular questions, either from the Q&A or from the
Thanks, Dan. Ricardo is actually going to take over now and ask a few questions of the panelists. So thank you so much for your questions and your time, Dan.
All right, thank you.
Hello, everyone. So yes, we have a couple of more questions. I think I will make use of the question coming from the QA, some of which overlap with the question that as organizer we propose also. So there is a question from Helena Kandarova, who is wondering how to deal with an MPS-based method for which no animal models were validated in the past. How to validate the most effectively, and I will add, would you consider an application if data were only generating MPS, whether there are no other animal models?
Ricardo, you're muted, but it looks like Donna has her hand up.
Yes, Donna, would you like to comment on this?
Sure. I mean, the FDA accepts all data. You know, when an application comes in, we don't say, sorry, this data we're not even going to look at. So look at all data. And I think the challenges in any new technology is if you try to replace a gold standard test, you get a lot of pushback. On the other hand, if you try to move into an area where there is no good test, then you're going to face these questions. So in my past, I worked on toxicogenomics, and I looked at idiosyncratic compounds. And in my naivety, I thought if a compound is positive for potential to cause liver toxicity, it would just cause a sponsor to maybe study ALT levels more often in the clinical study. But their problem was if it's a positive, what does that really mean? And there's no secondary assay to confirm or refute that positivity. So I think sometimes it's going to be very hard to move a new technology into an area where there isn't another assay, because then if you get a positive, the sponsor many times doesn't know what to do with it. So, you know, it's a tough balancing act between trying to replace a gold standard or trying to come in where there isn't anything. And I think it's going to be context driven. And again, I think a lot of times this comes to the level of the sponsor, not necessarily the FDA. But where does the sponsor feel comfortable putting a new assay in? When you get close to regulatory submission, I mean, most companies do not want to do anything novel because, you know, they're worried about what the answer could be and then how will they would explain it.
I think it's a great answer and very comprehensive. Thank you, Donna. And Sonia, I think.
Yeah, we like to concur. We still need to have indeed a big discussion on what is the gold standard, and there is a lot of going on around. Are these animal studies the gold standard, or do we need to really look at the clinical data and look at translational biomarkers that would demonstrate that an effect that is measured in a novel methodology, be it an MPS or another in vitro system, is translatable to the human situation. But that is a discussion we still need to have within the realm of the whole qualification exercise. What are the gold standards? What are we going to compare to, etc.?
Absolutely. I think I agree. Anyone else want to comment on this from the panel? Otherwise, I also have another question that comes earlier from one of the attendees, and actually I'm very curious too, as I also work with microfluidic system perfused with blood. And so I think we both, me and the other attendee, had a question regarding what would you say are the limitations of using whole blood into a microphysiological system? I gave my answer before, but I think he and also me would like to hear more about what you think about that. In my opinion, obviously, blood, it's a tissue per se, with the complication that when you perfuse a microfluidic system with blood, it's a bit like linking organs maybe, but with the complications to say that blood brings its own immunity. And so how would you consider that? And Donna, would you like to start commenting?
Yeah, I'm just nervous about putting any type of immune cell in a system unless they're coming from the same donor. So I've asked this question of some companies that have done this, but I'm still very concerned because you can get immune activation quite quickly. And I still would like people to explain to me how you're going to be able to do this with any kind of immune cell, particularly whole blood, in a system that's not concurrent with the, with the donor of the blood. Are you going to have cell systems from all different donors, and you're going to put an immune component in there? And what will that do? I'm really curious. I mean, I think we need a process to do this, but I'm curious how it would work and how you're not going to get immune activation.
So would you consider, maybe we open up Pandora box here, but would you consider a system made from an isogenic line, maybe iPSCs derived, and then combined with the blood from the same patient?
Yeah, at least, I mean, this is my personal opinion, but that would alleviate some of my concern. But I've worked a lot with the immune system and polymorphonuclear cells, and I'm just nervous about putting mismatched cells and blood together. And maybe the assays are short enough you're not going to have a problem, but if you have any kind of a longer term assay, I think you're going to have a problem, unless they're matched in some way.
I think we agree that in defined condition, control condition, which we can exclude sort of unwanted effect, that could be a good option. Using whole blood or PBNC, circulatory immune cells, could be a good option to modeling specific conditions, right? Any other thought, comment? If not, I think we'll need we can move forward, and I will leave then this virtual stage to Lina, who will announce the winner of our poster competition. Lina?
Yes, thank you. Yeah, as you've heard, we had a Young Investigator Award competition, and I wanted to emphasize that all the speakers which you listened today were selected from the abstract. So we have a great abstract submitted for this event, and we selected top to present today, and the rest of the abstracts submitted the posters, which you can look in the poster book. And with those abstract submission, we also announced the Young Investigator Award, and which will be the best poster will be granted the travel support to New Orleans in May, June 2022 to attend the first MPS World Summit. And again, thank you for attendees to vote for the best poster, which the voting was 100% in line with the Scientific Advisory Committee and Virtual Event Committee scoring. And the winner is Dr. Ilka Maschmeyer from the Tissues, who presented a poster on multi-organ chip development towards a paradigm shift in drug development. So congratulations, Ilka, on this award, and see you in New Orleans. Thank you. And with that, we are on the end of our meeting, and I would like to pass to Thomas Hartung, who will do the final closing remarks. From the car.
From the car. So this is the unique opportunity to open the meeting from Switzerland and to close it from Italy. I was listening into a fantastic day. I think we could not have had a better start to the series of events. I think we did the right thing to put the end goal first, talking about the regulatory side of acceptance of these models. Start the discussion how to meet these goals. We can talk about how these technologies can serve these purposes. And I think it was a fantastic program. I was really impressed with all of the contributions. There's more than 70 people to thank for all of this. Some making contributions to the science, others to administration, like Camilla did, and a wonderful committee organizing the virtual event. And I have highest expectations for the one to follow in December. I think we could not have started better. I'm looking forward to see you at all of these parts. I hope you agree with me. The future is MPS. I think it would have been wonderful if the animal experiments we want to replace would have received a critical stance like some of these models are also receiving now. But we want to be better than the animal experiments. And that's why it is good that we challenge them, that we have the highest expectations to these models. So hope you enjoyed. Please give your feedback. And I would like to close this. In this case, I don't have to wish you a good way home as I would have in another meeting. I hope you enjoyed the day. Thanks a lot.
Okay, with that, we will wrap it up. Thank you so much to everyone for attending. Thanks to the speakers, and thanks to everyone who helped plan.
Thank you, everyone. Great job.
Thank you. Bye.
See you in December.
Thank you. Bye-bye. Thank you.
Thank you.
Bye-bye.
Thanks. Bye-bye.
Bye.